Hi, everyone. I'm Nicole Kleinsteuer. I'm the Director of NICEDM and the Executive Director of ICFAM, and it is my absolute pleasure to welcome you to the ICFAM Public Forum 2024. This is one of my favorite things that we do every year, and we have a just tremendous schedule for you over the next day and a half. But first, I would like to welcome and introduce our new ICFAM co-chairs as of January this year. We have Dr. Suzanne Fitzpatrick from the U.S. Food and Drug Administration and Dr. Natalia Venus from the U.S. Department of Defense. Please.
So I'll be doing the first part, introducing everyone, and Talia will be doing the rest.
Yeah.
Thank you, everyone, for coming. This was really exciting to see everyone here and interested in what we're doing, and hopefully can partner with us. I know some of the things we'll talk about at FDA, we can partner with you guys on it, and you're the same, so, okay.
Yeah, we're very excited for this new chapter, and hopefully we'll keep moving forward.
So I'll start with introducing Allison Harrell, which she works with FDA on Talk 21. She's at ORD in Research Triangle Park, and unfortunately she wasn't here in person, but she will be here. And then we have to have someone find her. I see her there.
I'm just gonna move this over so everyone in the room can see the Zoom. Here we go. And Allison, if you'd like to go ahead and share your slides.
Sure. All right, there we go. All right, all set to get started, Nicole?
Oh, yes. Yeah, I think you're good, Allison. We may be having a sound issue in the room here. Hopefully you can hear me.
I can, yeah.
Okay. I think we're okay. Yeah, please go ahead.
Okay, great. Well, I'm really excited to be with you all today virtually. I'm so sorry I couldn't make it in person this time. But please don't take that to mean that we're not excited about this meeting and about the really rich partnership that we have with ICFAM and with NICEDM. So delighted to be here, at least virtually, to tell you a little bit about an update on our NAMS work plan implementation. And as Susie mentioned in the introduction, I'm with the Office of Research and Development. Specifically, I'm in CCTE, which is the Computational Toxicology and Exposure Center of ORD. We have 4 national centers and 3 offices that are organized to address a variety of issues. And we're based in Research Triangle Park in North Carolina. We share a large research campus with the NIEHS, and so we're really excited to have them as neighbors. We have published a large number of papers and journal articles in the last couple of years, about 223, many of which are focused on new approach methods and exposure methods, some of which I'll touch on, but given the short time frame, I'm just going to give you a couple of vignettes. But before we get there, I wanted to talk to you a little bit about our NAMS work plan. So this is predicated on the toxicity testing in the 21st century vision and strategy that came out in 2007. There's been quite a lot of work to get us here, but in 2021, the EPA released our new approach methods work plan, and that work plan was organized around 5 deliverable areas with the idea that we would deliver on a number of short-term goals by the end of 2024. And so what was included here was to develop new approach methods to address information gaps, engage and communicate with stakeholders, establish scientific competence and demonstrate application of NAMS, develop baseline and metrics for animal use within the agency, and evaluate regulatory flexibilities that enable us to utilize information from new approach methods for some of the regulatory decisions that are made agency-wide. And so our NAMS work plan represents a snapshot in time and will evolve as EPA's knowledge and experience grows. So I'm going to give you a couple of vignettes on deliverable 1, which is developing and implementing NAMS. We have quite a large portfolio of work in this area. And so what I wanted to highlight here is a case study being led by Katie Paul Friedman, but being done collaboratively on an international stage under APCRA, which is accelerating the pace of chemical risk assessment. And so what this group has been working on is integrating new approach methods inclusive of bioactivity-based methods and exposure methods in order to gain some insight into prioritization of chemicals for further testing. And so what was done here was they selected 200 chemicals that have been tested in ToxCast. These represent a range of phys-chem properties, commercial use cases, and quality control was done on them. There's some QSAR information that has been collected using a variety of models, and then bioactivity measures inclusive of cell painting or high throughput phenotypic profiling, high throughput transcriptomics assessment using a variety of different cell lines, representing a range of biological coverage. We also have targeted testing, including adagene assays, biomap assays, in vitro pharmacology, neurological testing using microelectrode arrays, some DevTox, as well as ToxCast ER and AR models to look at endocrine disruption. And then in parallel, there are exposure methods that are being brought to bear to consider where the threshold of predicted exposure might be close to a point of departure that's derived from our NAM models. And so that's what's highlighted here on the right-hand side. These are some data from a forthcoming publication that's being developed at this point in time. And so what you're looking at here is the point of departure, the dose, at which you see bioactivity, and that's in green, purple, and blue. And then the predicted exposure by a variety of models, which is the more yellow. And so where that point of departure is getting closer to the exposure case, that's where we might want to do a deeper dive and figure out, is there indeed a human health assessment that should be done in more detail on these particular chemicals. So that's just one way in which we're taking these very discrete assays that we've developed and starting to package them together and thinking holistically about how to use this information to gain insight on specific chemicals that the agency might be interested in. So again, stay tuned for that upcoming publication. In addition, we've also been working on a variety of new methods for assessing exposure. Tools for bioactivity assessment are complemented by assessing exposure, and we need a variety of methodologies in order to do that. We've invested quite heavily in non-targeted analysis. The reason is that targeted analysis methods are trusted, and the inclusion of standards aids identification, but these methods are not amenable to large-scale measurement or surveillance of chemicals in a variety of environmental media that are of interest to the agency. And the other issue that feeds into our desire to develop NTA methods is that chemical inventories are growing and there's increasing concern on how to tackle mixtures, degradants of these chemicals, and their metabolites. So I'm not going to go into a lot of detail on this slide. This is really just to highlight some work that's being done by John Sobis and Alex Chow. But essentially what's being done is development of NTA workflows, some of which have been published, that allow us to consider appropriate ways to acquire the data, process the data, and then identify which chemicals are associated with the various peaks that come out of the NTA analysis. And in order to facilitate this identification, John Sobis and his team are developing what's called the NTA web app. And so there's been some scoping of this already in which we're able to import data into the tool and begin to assess what degree of confidence we have in peak to chemical assignments so that we can start leveraging this information to assess which chemicals are present in environmental media. So that's another tool I just wanted to highlight for you that is soon to be coming online and is actively being worked on. So then moving into deliverable 2, which is our training engagement piece, we've developed a NAMS pilot training program. Again, this is one of our key deliverables under the EPA work plan. for NAMs, and we started this back in 2022. We've had a number of different tools where we've offered training. Initially, this was online due to the realities of the COVID-19 pandemic. So we had training on ecotox in May 2022 and again in 2023. We've had trainings on the complex chemicals dashboard. We've had trainings on GENRA, which is one of our read across tools, as well as on HTTK, which is high throughput toxicokinetics that enables IV/IVE analysis of in vitro derived information. And then just this last April, we had an in-person training for a couple of days. We had about 88 registrants, about 20 to 25 of which were international folks. So that was pretty exciting to get a very diverse pool of folks on campus to talk about a variety of tools. And here we did a survey of tools just to get people a flavor of what's available and how to get started on using these various information streams for their particular use cases. So we covered the AOP Wiki, Chem Expo knowledge base, Chem Informatics modules, GENRA again, ecotox, SeqAPass, which is essentially looking at sequence homology across a variety of species for ecotoxicology applications, ToxCast, APIs for computational toxicology and exposure data, and other sorts of NAMS resources, like giving them a flavor of some of our high throughput testing modalities like transcriptomics and cell painting, for example. So we're continuing to offer these sorts of trainings and plan for the next one in the sequence. Another area that we've been working on very actively, now moving to deliverable 3, is to collaborate with our partners in the field to build confidence in NAMs. But this really requires frameworks to assess how to build confidence. So many of you have an awareness of this publication that came out a couple of years ago, where there was heavy NICEDM and ICFAM involvement, and EPA was involved in this too. Building on this framework, I'm sure you'll hear more about this at this meeting, but just a month ago, ICFAM released its validation, qualification, and regulatory acceptance of new approach methodologies framework, which really was built on a lot of these earlier established principles, but went into a number of additional details on how to design experiments effectively and around some of the US interagency federal partnership thinking around this idea. So we're quite excited that that came out. And then building on these frameworks are a couple of international and EPA activities. One of them is a revision of OECD Guidance Document 34, which is around how to validate test methods for use in hazard identification. And so the US is co-leading. Myself, Nicole Kleinsteuer, Warren Casey, and Charles Kovach are the US leads of this. And that effort is underway and having a lot of very productive conversations between countries on, how do we establish confidence? How do we modernize workflows for validation of NAMs and all of that sort of thing? And then related to that, EPA is developing its own confidence framework. But importantly, our confidence framework, which is intended to be delivered at the end of 2024, is going to be harmonized with all of these related efforts. So stay tuned for that. And then deliverable number 4 is developing baselines and metrics for animal use. Part of what's motivating this goal was a GAO report to Congress that came out in 2019 that recommended that federal agencies develop metrics to assess progress toward reducing, refining, and replacing animal use in testing. Our 2021 NAMS work plan established goals to quantify animal usage for R&D and also for regulatory testing requirements. And so initially we're starting with mammals. We have a stretch goal to be more inclusive of all vertebrate animal testing in the agency because we have a lot of eco-species like fish and amphibians, for example. OPPT and ORD are now publishing annually metrics on animal use, and we've established baselines for mammals. And again, this process for including all vertebrate animals is ongoing and something that we're working on very actively. So you can check out these web pages. Just highlighting here just a screenshot from our colleagues in OCSPP, and you can find when you go to this web page a table looking at regulatory waivers that have been issued for a variety of different testing modalities. And then number 4 is evaluating regulatory flexibilities. So we have drafted a document that assesses regulatory requirements and flexibilities to use a variety of information sources. Incorporation of new scientific approaches into regulatory decision-making is an iterative process in which several statutory and regulatory barriers have to be considered. And what we found in doing this assessment is that although the majority of EPA's statutory mandates do not specify the types of testing the agency must require, language across statutes generally indicates that the scientific information considered should be of high quality, based on scientifically sound methodologies, and be subjected to peer review. And so within this document, we're going to be discussing specific statutory and regulatory flexibilities to utilize alternative methods and information sources. So this is just kind of a commercial right now. We've got this document in draft and it'll be soon going into clearance, and we hope to release this document sometime this year. Okay, so that brings us to the end of updates on the NAMS work plan. So the first 2 deliverables are more or less complete. Of course, we're continuing to develop NAMS. And the last 3 are ongoing, but certainly we have plans in place to complete these deliverables by our stated deadline. So fingers crossed on that. And we'll be continuing to keep working on this, and we look forward to giving you hopefully an update next year saying we're done and we've achieved victory, and we can think about kind of the next few years ahead at what the work plan is going to entail. So leaving that behind, I wanted to talk to you a little bit about an effort to update ORD's portfolio of human health assessment products. And part of this is motivated by the challenge that there's a lot of chemicals in commerce that have no toxicity data. You can see in the 1984 NAS report, this was highlighted, but it's only gotten worse over time. So a 2020 survey of 19 countries and regions found that there are 350,000 chemicals and mixtures of chemicals that are registered on one or more inventories. And the plots that I'm showing you here on this slide are various buckets of chemicals that we're interested in from an agency standpoint. So on the left-hand side, these are chemicals in these buckets that have toxicity information. And on the right, these are chemicals that have a human health assessment that has been issued on them. And the colors here, without getting into too many details, these are types of tests that we have data for, or types of human health assessments that we have information for, given our different regulatory statutes. And what you can see is that, few chemicals in these buckets have toxicity information. And when you look at the human health assessments, fewer than 20% across a variety of chemical inventories that we're interested in have an associated human health assessment. So we really need to introduce efficiencies where we have high confidence in doing so. And backing up, the other thing I wanted to highlight here is that the time and resources that it takes to go from the chemical testing phase to the assessment phase can be quite long, depending on how complex the assessment is. So for traditional animal studies, so thinking about 2-year bioassays, the time that it takes to finalize the information from that study can range anywhere from 2 to 10 years. Developing the associated chemical assessment can take 4 or more years. And so you're looking at a very long runway going from no data to having the human health assessment issued. Thinking about that, ORD has been looking at our portfolio of human health assessment products, and we have very high quality assessments, ranging from things like our integrated science assessments and our assessments issued under the IRIS program that require a large body of information in order to inform the development of the assessment. So these are done on data-rich chemicals, as we call it, and they can take they can take a hot minute to put together because you have to evaluate the body of literature, assess study quality, and then make a determination on the overall weight of evidence. We have a couple of products that are sort of in this middle ground of being between data rich and data poor. These include things like PPRTVs or PALs or some of our new human health toxicity assessments. But until this year, we really didn't have anything in this space for human health assessment development of data poor chemicals. And one of the opportunities that we have here lent itself to development of a new EPA transcriptomic assessment product in which we can gain some information about a bioactivity-based point of departure and issue a human health assessment on the data gathered from that short-term study in as little as 6 to 9 months after chemical procurement. So we've developed that, we've published on it, and we're utilizing that paradigm now. We're also starting to think about how we can utilize more new approach methods to assess bioactivity and hazard-based assessments in a variety of different contexts. And as part of our weight of evidence for this, we've developed a value of information framework to assess the trade-offs between the uncertainty in the data derived from the assay, the cost of the assay, as well as the socioeconomic costs that are borne by society, and then time. So time being how long does it take to actually develop the assessment, and also considering a time horizon of exposure to a potential population who might be exposed to that chemical in the environment. And so in our case study, we considered a short-term assay that took 6 months to develop with greater quantitative uncertainty. And so this is looking at reproducibility of the point of departure from run to run. And then the costs were estimated around $200,000. And we compared that to a variety of information we had on a 2-year bioassay, which on average takes about 8 years. The uncertainty is lower in part because the database is much greater. So we have a lot of information there. And then the costs are on average about $4 million, although I will say we did sensitivity analyses looking at lower cost here, as well as a shorter time frame to develop the assessment if we're being optimistic in that development. And so part of our motivation here was that the national academies recommended that VOI analysis be used to make these sorts of determinations for assessing the value of using an alternative approach versus an established approach. And I just put this quote here from Eckhart Beck from Region 2, thinking about sort of the time frame of Love Canal. If health-related dangers are detected, what are we as people willing to spend to pick up to correct the situation? How much risk are we willing to accept? And so this sort of framework helps us to contextualize risk with cost and health benefits to the exposed population. So I won't go into this in too much detail because I don't want to take up too much of your time. You can find our draft VOI analysis on the web. I've put the URL down here to our draft analysis, and the final version of the analysis after we got comments from Board of Scientific Counselors will be forthcoming in the next month or so, we anticipate. But essentially what we did here is we looked at our 2 testing scenarios and their associated human health assessment processes. We had 18 baseline scenarios where we continued varying levels of exposure for the exposed population and variability in that exposure. We considered a couple of different risk-based decision contexts, so target risk or benefit risk. We have 342 sensitivity scenarios. So what if the population size is more or less that was exposed? What if the cost of testing was different than our baseline assumptions? Looked at the uncertainty variability, the quality of exposure information, the time horizon of being exposed, whether that was 20 years or more or less. The time required to complete the testing and assessment was shifted less or greater. We looked at more or less health effects valuation. So what is the annualized cost to that individual for seeking health care to treat it's sort of maybe a more minor scenario versus a more major scenario. So thinking of something like asthma versus maybe something that was more debilitating, for example, depending on the health effect of the chemical, and then the target risk level. And out of this, we get a variety of different VOI metrics, including return on investment, cost of delay, expected net benefit of sampling, and expected value of delayed sample information. And what we found is that if you consider the benefit risk decision context, 83% of the scenarios favored the shorter duration assessment, even though the quantitative uncertainty was a little bit greater than the conventional test. 17% of the scenarios favored doing no testing at all because the risk was so high that we could go ahead and make a decision without more information. And then in the target risk decision scenario, 87 to 99% of scenarios favored the shorter duration assessment, and 7% favored doing no testing at all. So this provides a really nice framework for us to contextualize those trade-offs between uncertainty, cost, and time in the context of socioeconomic benefits to society. In addition, we've been working on some studies to figure out how to benchmark NAMs. So how do we contextualize uncertainty and reproducibility for a NAM in view of reproducibility and uncertainty estimates that we gain from animal testing scenarios. And so Katie Paul Friedman in a past study leveraged information from Talks for FDB. I'm not going to belabor this because I think I'm running a little over time, but you can certainly pull her paper from 2020 to take a look at this and check out her estimates of root mean squared error and what that might mean for contextualizing uncertainty of NAMs in the future. And then in parallel, we've been working on an APCRA case study leveraging information from the Pharmapendium database, which looks at pharmaceutical therapeutic drugs. And we've started looking at concordance in the overall point of departure between humans and rodent models, as well as rodent to rodent models. We're seeing in our initial analysis a pretty low concordance in just looking at overall point of departure. And we're doing some follow-up studies to look at some very specific hazard-based concordance estimates. So that's a case study that is ongoing, and we'll look forward to reporting more concrete information from that in the future. So with that, I'll wrap up and say work is ongoing to complete our deliverables under the NAMS work plan. Our VOI analysis helps us contextualize the trade-offs and uncertainty in NAMS with the return on investment and socioeconomic public health benefits realized by reducing the time to a regulatory decision. And I'm really cheered to see that others in the field are starting to utilize our framework for other contexts. So I hope that people get some more insights out of that and are able to utilize it moving forward. However, there are barriers to adoption of NAMs for regulatory contexts that still necessitate confidence building through our partnerships with ICFAM, including development of case studies, adding details to assay validation frameworks, as we're going to do in our GD34 update, and consensus building and peer review. And with that, I will stop and turn it back over to the moderators. Thank you.
Thank you so much, Allison. That was a tour de force of all the amazing work that EPA is doing in NAMS. We do have time for a couple questions. First, I want to make sure that our captioner is fine. I see that you have your hand raised. I just want to double check that everything's okay there. You don't see anything? Okay.
They should be fine.
Okay, just making sure. Okay, so let me pull this over. And first, I'll ask if we have any questions from folks in the room here. Nope, not seeing any hands raised. So we'll go to the questions online. So the first question online from Dr. John Gordon at the CPSC is, do you have an idea of when the web app will be available or possibly demoed? Looking at the timestamp on that, I'm guessing it's on the non-targeted analysis web app.
Yeah, John, that's a really fair question. I'm glad you asked it, but I have a very unsatisfactory answer because we're still working on some of the underlying processes computationally. Um, it's something that's under development, but it's not quite ready for prime time. And I think we need to do some beta testing before we can actually come up with a firm date when we're going to be releasing it. But I tell you what, John, what I'll do is I'll send a note to John Sovis and see if he can give me an estimate. And then I'll send you a follow-up email. And for any of those folks who want to know about it, I'll copy you, Nicole, in case others have the same question.
Perfect. Thank you so much. Okay, our other question online is, in 2019, the EPA announced plans to eliminate the use of mammals for chemical testing by 2035. They have since seemed to abandon this plan without providing details on that decision. Can you speak to that decision to abandon the plan and whether a future target date has been discussed?
Yeah, well, I'll tell you where I'm sitting in ORD, I really can't comment on EPA policy. But what I will say is that we have a really strong commitment to developing and utilizing new approach methods as evidenced by our NAMS work plan and the deliverables that we've outlined there, which are really aggressive. So I would just reassure that we are working quite hard to be able to utilize this information in-house and really looking forward to some of the advances that are coming both in-house and from our partners in the field to be able to modernize our approaches and utilize the best available science.
Okay, thank you very much, Allison. And I will note for folks online that there are several other questions that have been answered in the chat specific to logistical questions around whether or not the slides and the video will be made available, which the answer to that is yes, once we get the 501(c) or the compliance. We have a question in the room, 508. Those are the numbers I was looking for. Thank you, Emily. We have a question in the room before we move on. Can you do the microphone for our friends online? Thank you so much. Make sure it's on. Let's make sure it's on. Let me see the green light. There you go.
Special button that you have to press. Andrew Rowan, Wellbeing International. It's been a while since I've looked at the legislative language, but I seem to remember that TSCA required a rodent LD50 in some of that language and for Superfund sites. And I'm just wondering how EPA is addressing that particular issue.
Yeah, well, two answers. One is I really can't comment on regulatory policy. And two, I'm not, honestly not the best person for this question, sitting in the Office of Research and Development, which I know is not satisfying as an answer. And I wish some of our regulatory colleagues had been able to attend this meeting. I know a couple of our folks who usually sit in on these are on international travel. So unfortunately, I don't have an answer for you. But what I can do is I can write it down and see if we can get some follow-up on that question for you. And maybe that's the best I can do today. So I'm sorry about that.
And I thank you, Allison. And I will add to that in the NICEDM update tomorrow, I will talk briefly about a collaboration between NICEDM and the EPA to evaluate the use of the CAPMOS consensus QSAR model for predicting LD50 and the performance that it demonstrates against, in this case, it was technical grade active ingredients, but there's potential, I think, to extrapolate from there. Okay, one more question in the room and then we'll move on.
I'll make it quick. Hi, hey, Allison, how are you? Sean McCullough, RTI International. So you did a great job of describing the interactions between ORD and OCSPP in terms of furthering the reduction of animals and increase in NAMS use. What is the engagement level with some of the other program offices that look at regulatory decision-making quite a bit differently than OCSPP, yet still rely, at least theoretically, on animal data to regulate things like hazardous air pollutants? While the number of agents is significantly smaller, the number of, especially Americans, affected by exposures to HAPs are easily in the millions. Can you give us some thoughts on that?
Yeah, thanks for your question, Sean, and very astute as a former EPAer. So I would say, we're actually working very closely with a number of our regulatory partners. Obviously, with a short talk like this, I wasn't able to get into all the areas, but we have a very close partnership with Office of Water, particularly on some of the NTA work that I mentioned, where they're interested in exploring use of that technology for environmental monitoring. There's also very close linkages between one of our center's component groups in the Office of Water, which is the Great Lakes Toxicology and Ecology Division, which is very much focused on eco-tox, where they're evaluating a number of different tools to advance sort of new approaches in that space, including high throughput transcriptomics analysis using models like the RT-GIL assay, for example. So there's an awful lot of work going there. And we also have some discussions ongoing with OLEM on utilizing some of our tools for some of their regulatory context of use. In addition to that, we also work really closely with our regional partners. In particular, we have a ROAR project on again, looking at NTA technologies as well as other sorts of tools. So we're working very closely with a number of our regulatory stakeholders, and we really enjoy their partnership to ensure that the data and tools that we develop in ORD are useful in the regulatory context. So thanks, Sean, for that question.
Thank you so much, Allison. Okay, so we're gonna need to move on in the agenda here. So our next presenter is Susie Fitzpatrick, and she is from the US FDA. Oh, don't worry, I'll do it for you. She's from the US FDA Center for Food Safety and Nutrition. And I am going to share her slides. Here we go.
So thank you very much for inviting me here today to talk about the programs we're doing in the FDA's Human Food Program to advance new approach methods. And we're really doing a lot, and we're very excited in order to share it. Let's see if I can go forward. Just to remind you, the Center for Food Safety, and we're undergoing reorganization, so it'll soon be the Human Food Program, has a lot of compounds that we look at. Not only do we look at food additives, packaging, grass substances, cosmetics, dietary supplements, contaminants, which are like metals, constituents, which are like heat process contaminants, and macronucleates. So we have a lot to look at. And the contrast between the Human Foods Program and our medical product centers is that for the most part, ours is a post-approval post-approval program. Okay, what that really means in regulatory terms, if it's a pre-market application, then the person bringing that application in has to demonstrate safety and effectiveness to the FDA. But once something's on the market, the burden shifts over to FDA to demonstrate harm, however that product is used. And that is a big burden to do because, we could be taken to court and we can be sued, et cetera. So it's a very big burden. And that's, I think, why we feel that for FDA, new approach methods is where they're really needed more than any other part of the centers because we don't own most of the things. Nobody owns or has any ownership of most of the chemicals in food, and we have to show harm before we can try to remove them from the market. And we know, we're not saying that animal studies haven't given us good results, but we're saying that if there's something in the marketplace and there's a few that just recently caused some liver toxicity, it's really not acceptable to tell to our consumers, like, we'll be back to you in a couple of years after the animal study is done to tell you what you just ate and gave to your children is harmful. So we really need some methods that can give us some indication of what's going on quickly to identify the toxicity in a more timely method. And that's why we put a lot of resources into our new approach methods at CFSAN. So what are we doing? We have activities divided into 4 areas. We have center activities and partnerships. We have agency across the FDA agency and partnerships, cross-agency partnerships, and a lot of global activities and partnerships. One of the things that we did, let's see if I can get this for it, recently is and this only applies to food additives and foods, is we published a paper, The Use of Dogs as a Second Species in Tox Testing Anywhere, saying we would say that instead of putting a dog for a second species in a food additive or packaging thing, not for any other part of FDA, we might say you might want to substitute that dog study with a new approach method that will give us more information about the mechanistic ability of that. We don't have any guidance or regulations that say you have to put a second species, a non-mammal second species in there. So this was one place where we found was easy after reviewing 160 dog studies that we have in our, that this is a place where we could easily put an alternative. And so the paper was published a couple of years ago, and the reference is right there. So what have we done at the CIFSTEN level? We've developed a NAMS working group with representatives from all parts of FDA. Understand, I'm a regulator, not a researcher at CIFSTEN. We conducted a survey of the food industry to see what the food industry is using now, according to NAMS, and mostly they're doing in silico methods. Then we conducted a gap analysis through all of our programs to see what new tools we wanted by CIFSTEN and where new approach methods could fit in. And this gap analysis then will help us direct NAMS research with our stakeholders. So that means we looked at the context of uses that we might want to be developed for any research. And then we want to encourage partners to partner with CIFSTEN to work on these. So if you look at the FDA website under like CEDAR, Science and Research, CDRH, Science and Research, and now under CIFSTEN, Science and Research, that's where you're going to find the new approach method and Office of the Commissioner. So we're going to pretty soon put up our gap analysis, but also ways to partner with FDA. You have research collaboration agreements, which are fairly easy to do. CRADAs, where you bring technology from other places into, and we have CRADAs, for example, with the Emulate to bring some of the chips into our labs. We can do material transfer sheets. We can do memorandums of understanding. There's a lot of ways that we can work to partner with our other people to work on new approach methods. So that's what we did. We took a leadership role here, and we wanted to focus on how we're going to qualify some of these methods too. We're not just doing the research, but we want to look at a qualification program in the foods part, because qualification for drugs is very different than qualification for foods or devices. They have different uses and different criteria. And one of the things we're starting to look at is what we're tentatively calling a program as Entree. And it's to really look at the specific context of use at FDA drives the extent of information that you need for a qualification. So if it's for a screen, you don't need as much data to use that as a screen. For a pivotal study that replaces something that you ordinarily ask for, you're going to need a lot more. In other words, looking at it, what's the consequences of a wrong answer? So a wrong answer for a screen is not nearly as serious as if you use a new approach method in place of a pivotal study. So we want to look at our program. We're trying to design it around two sort of tasks, and we're working on these little bubbles right now. But the first part is the biological relevance. And I think this goes with what we've said in the ICFAM new guidance is we want to say, is this really relevant? Does this really show what's going on in vivo? And if you're not to the point where you can say this is going on in vivo, that you have enough confidence, reasonable certainty that you have confidence that this actually identifies, measures, or predicts what's going on in vivo, then you stop right there. But after that, if you do feel that way, then you can move on to technical characterization, which is using the context of use, the things we've seen, established performance criteria and acceptance for different NAMs. So this is the program we're working on now, and also want to do some test cases for it and then put those on our website. So we also have a program we call Training by Doing. Okay, so Training by Doing, we want to consider succession plans so all of us feel comfortable retiring and not leaving everyone not knowing anything. So we call it Training by Doing. We're putting our junior scientists working on areas where they actually wouldn't be ordinarily on senior projects where we're giving them a lot, including like Entree. We have 2 junior scientists really working to help us identify that. And then working with biomarkers, working with all the different committees that we have. And then they get the opportunity then to talk with the senior management or some of our global partners on this, which is an opportunity I know when I was a much more junior scientist would have been really exceptional. Now we're probably nervous if that happens to us. We have to go in and talk to the senior people. So we're really trying to make sure they feel engaged and comfortable to take over these places by giving them more and more responsibility, including to their regular job. And that's really been successful because they also have more time to do things than we do. And so, so it's really been a great program for them. We've also done some workshops that have been co-chaired by San. We co-chaired the first MPS Summit meeting that was in New Orleans. We co-chaired a global meeting of ILMARAC, and I'll tell you what ILMARAC is, our global regulatory food safety regulator group in Parma in September. We worked with NIEHS and JHU to do a model to do a trust your gut model to look at different intestinal models, and that's about to be published in all texts. We also did a workshop on assessing developmental neurotoxicity, which is also about to be published. And we want to do a global workshop. We were planning on doing a global workshop on MPS for food chemical assessment. One of the FDA started the very first program on microphysiological systems with DARPA and with NCATS when I was in the commissioner's office. And so a lot of things that we're asked at CIFSAN is how do I use MPS in the food chemical area? And that, you know, most of the you know, attention's been how you use it for first in human or demonstration in the drug. But that's a small, narrow area, but food chemical or chemical risk assessment is a much broader area. And actually, we've talked to, they're having a 4th World Congress on MPS in Brussels in 2025. And we talked to Matu Bitkin, who's running it, that he have a large section now on chemical risk assessment. And you'll see some other things that we're doing on that too. And then we have a meeting of our big ILMARAC is going to be in Berlin in December. So okay, one other, let's see, now I'm not even being able to do this. Okay. Go on. Okay, you wonder if I can even do any names if I can't even work this. Okay, there we go. So one of the things that we're also doing is we are working on a neuromuscular junction for Botox testing. So we are working in our lab. We have partnered with DITRA on this. We've partnered with a company in Canada on this. And we've also partnered with NCATS to put out 2 solicitations to develop, and we're working with our Arkansas Regional Lab, which does the mouse lethality testing for food and for drugs too. So we are thinking that this, and we're very close, this is a really exciting project, and we feel like it's going to be the first microphysiological system to be qualified because you don't need to show human relevance to this chip. You only need to show that it's good or better than the mouse lethality test. So we're working very hard on this in our lab and with our partners, and I think pretty soon we're gonna be able to present on that one too. Okay, so let's see if I can do this now. Now, where have we partnered? As I said, we recognize that we need to work with we don't have a lot of the research dollars that maybe some of our federal partners do. So we're really relying on partnership, not only for research dollars, but for expertise out in our stakeholders, because to be honest, all stakeholders want good, safe and effective food. And so everyone is willing to partner with us to try to get some new tools in our toolbox to make sure that happens. So one of the things we've done, as I said, we've developed partnerships on the neuromuscular junction. We just sent an RCA with a CAT to work on them, and you'll see we have a CERSI with them too, and we're working on that. We just signed something with Robin Tanguay to look at what they're doing in zebrafish. We are going to sign an RCA. We're meeting with Unilever next week to talk with them about it, and we also have We also just had bilaterals with EFSA to work together jointly on a lot of the products for NAMs. So we've been very busy developing partnerships to do that. So now also FDA has an alternative methods work group that I chair with Dr. Shi from NCTR, and that discusses activities across FDA. And one of the things that we've done is we have this webinar series that we've been running for about 4 years, where we ask people if they want to come in and give a seminar on a new approach method that they have that has to be fairly advanced, so it has some sort of context of use and some data. So we're not really trying to help them. We have other ways FDA can help people, produce their method. But this is a seminar series. We actually were really excited because, I know some people were promoting that book Rat Trap, which wasn't very complimentary, except for our seminar series, which was really a surprise that they said this was really nice. And then sometimes we adopt some of the methods that come in and help bring those forward. And I know our center has adopted a couple of them too, to try to then work with those developers to bring that method forward so that we can be using it in our programs. We're trying to now expand this program to now, instead of just waiting for people coming to us, now we're looking for people to give seminars to. We looked at the people that just got the ARIES grants, and I sent a note to one of them inviting them to present to FDA. When we read the literature, we're seeing people that are developing methods in the literature that we want to bring to FDA to talk to them about it. So it's a really good way of all of FDA being up to date on some of the things going on. And it's not just CIFSAN that works on them or invites them, it's all of our centers bring different seminars to us and bring us ways or topics. Recently, our CBER people brought in the Korean Institute of Technology, and Chad is working on a global MOU with Korea to work with us on across the centers on that. So that's been a really great program for us to invite people in and really keep up to date on the literature and what's going on. So one of the things we also did with our alternative methods work group is we developed a CERSI, which is a Center for Emerging Research and Science Innovation with Johns Hopkins for their mini brain. I'm sure people have seen Thomas present the mini brain, and we're using the mini brain to look at mixtures of metals and developmental neuro. And so we've talked Thomas into being our first guinea pig to try out our qualification program, our draft qualification program, Entree, on his mini brain to see how we can develop the criteria for this particular thing and move it forward. So as most people know, most of the regulatory parts of NAMs use case studies in order to really try out things and put them on the thing. And that's what CIFSAN is trying to do too, is case studies. So any other volunteers for case studies, we certainly would consider that. Just to mention, I said we work across the agencies. We do work with CIFSAN represents FDA on Tox 21. Obviously, we work on ICFAM. We co-chaired the new qualification program that came out. We also co-chaired the metric program to show our real support, especially in the foods area, for ICFAM and the work that's going on there. And this is also another thing going on. People asked recently about non-targeted analysis and evidence-based monitoring. There's a big program under OSP, the Office of Science and Technology Policy, to look at contaminants of emerging concern and how we can use new approach methods. And Chad, that works with Warren and Chad Lifestone, and I chair the contaminants part, and NIST and EPA chair the non-targeted analysis and evidence-based monitoring. And they just had a big seminar on non-targeted analysis and across the agency. I think they had about 180 people, and then they're going to put that up on their website. We are looking at, as a case study, trying to put some of our criteria for chemical assessment on a compound that's in water, but that seems to be everywhere now. 6-ppd quinone coming off tires and being in there. NTP has taken the lead on that, but that's really cross agency federal program, which is what out of the White House, we've been told we have to work cross-agency on some of these things that affect more than one federal agency. So that'll be an interesting way and see how it works. And we're also under our contaminants emerging concern, want to have a series of workshops on communication to make sure the federal agencies are all communicating together and saying the same thing and communicating to the public and to learn how to communicate, and especially to diverse populations for some of these things. So that's something else that should be coming out soon from OSTP. They have two other working groups. One was on PFAS and one was on sustainable chemistry. Those are a little farther behind the contaminants of emerging concern, but there's still ways of us working cross-agency on that. Let's go next one. So we really feel in CIFSTAN that global partnerships are what really advance the methods for chemical risk assessment, and they really improve confidence in the consumers and stakeholders, and that's global partnerships with regulators in mind. So we work with ASPIS, which is the an international program out of the EU, which is Risk Hunter, OnTox, and Precision Tox. So CIFSTAN represents FDA on that. What we're doing with Risk Hunter is we're working with them on the tissues chip. And we're also hoping that they have a monitoring algorithm, ASPA, which keeps track of all the risk assessment, and they may give us one of their, at least a copy to us about that. For OnTox, we're just about to sign an RCA to use some of the methods in OnTox that looks at mixtures of a chemical, a product that was on the market and caused quite a bit of toxicity, a surprisingly benign project that put about 500 people in the hospital. And then we also represent, so that's ASPIS and Precision Tox. They're looking at biomarkers of lower organisms that then could be used for human risk assessment. And we have a lot of data on C. elegans, and I just talked to Allison and John Wabach. Could we develop PODs from C. elegans data and see if that could be used as a rapid screen? We're looking for a rapid screen to quickly tell us where we should be looking for all the chemicals that you'll see in the food. And then we also work with PARC, which is a partnership for accelerating risk assessment in chemicals. That's a very big area that we just had a meeting with them on it. And then we developed what's called the International Liaison Group on Methods for Risk Assessment of Chemicals. That's all regulators from different countries. And you can see it's really to develop risk assessment methods across the globe, to include food regulators in new approach methods, to have action plans on harmonizing these methods, and to facilitate global harmonization, joint events, projects, and exchange. As I said, we just have bilaterals with EFSA, but this is a lot bigger. And ILMARAC is actually FDA's biggest global coalition that goes on, and you'll see in the next slide. This is ILMARAC. And since we had this meeting, 3 more countries want to join because this was shared. We started this with EFSA, but now BFR took over. That meant we can include the UK now in this, where before EFSA wasn't keen on that. Switzerland and Norway. So this is our ILMARAC meeting. We meet 2 or 3 times a year and have one person session that we had in Parma. So one of the things we're going to look at, we can look at draft mandates, guidance, options, themes, methods, models, chemical databases, emerging topics, just a whole standard of things we can use using this partnership, which everyone is really very eager to participate in. Go on to the next one. They're all, as I said, governmental and intergovernmental food safety regulator organisms. The technical secretariat rotates every 3 years. So now they took over BFR, which is Germany, took over from EFSA. They are really tough. I mean, they made it really much harder. We were just meeting with them last week, and they're very tough. And then we have meetings, like I said, 2 or 3 times a year, meetings in person 1 or 2 times every year. And we're meeting in Berlin in December. So what are we doing? Oops, let's go back here. So what are we taking over? We are looking at 3 priorities. We're looking at mixtures, we're looking at human relevance, and we're looking at MPS for chemical risk assessment. So what are we doing with MPS for chemical risk assessment? We're planning a ring trial with Tissues, which is the German company has the chip, BFR, EFSA, CIFSAN, and NCTR, Singapore. And the Tissues chip is in a lot of other places too, and we might ask them to join to look at MPS and chemical risk assessment. So and EFSA, we're planning on putting this all together. We're just starting the planning once a month. Everyone's agreed to this. We have the chip already. BFR, obviously Tissues has the chip. Singapore is working with us to put the chip in their laboratories. BFR has just bought the chip too, and we really want to see, and then we will be making, our NCTR lab will be making the mixtures and then putting them out to everyone. And then we'll be talking about this in Berlin. We'll be talking about this, as I said, at the MPS World Summit, 4th World Summit in Brussels. And we're also talking about including in Ilmarak discussions with Park on chemical risk assessment. And I think that's all I have. Let's see. So actually, moving forward, the food program, we don't have a lot of resources, but we're making the most of the resources that we have and the partners that we have here across the federal agency, across the FDA, the federal agencies, and globally to really bring new approach methods forward, because we understand the urgency and importance for the food program of having some of these options and to assure that the things that you see in food are safe and effective. So with that, I'll thank you. And if you have any questions. No question.
All right, can everyone hear me? Thank you, Susie, for that excellent question. First, do we have any questions in the room? Not seen anything. First question is in the chat from Ellen Berg. Who are the collaborators on the neuromuscular junction Clostridium project?
Who is the collaborators on the neuromuscular junction on a chip? Okay, it's our lab, Sashi Sharma. Okay, and he's been at the World Congress on Alternatives, and so he's the lead on that in our laboratory at the Foods Program. He's worked with Curie Bio is one of the people that got one of the things. Hesperus got another one. With those are the two people that got the thing, and he's working with Canada, and I shouldn't even remember the name of that. Canadians, they couldn't apply for the NCATS grants because they're not US. And he was working with Ditra on it too. So he's got a lot of different people that are working on the chip with him. And then of course the Arkansas Regional Lab, which does some mouse lethality tests. We want them to be there as we work on the neuromuscular junction chip as we're working on it. So they'll be able to help us implement it and make sure that it's going to really replace the mouse lethality test. So they're side by side with us, as well as our Office of Emerging Terrorism, Counterterrorism, that kind of thing, from the Office of the Chief Scientist, because we feel like it's not maybe just for botulism toxin testing, but maybe for all toxin testing. And so we want to make sure that they funded us and also get them involved too. So, and if anyone is interested, I'm sure Sashi would be more than happy to talk to you about it. So.
All right, thank you. Next is from Christy Sullivan at IIBS. She says, thank you for this presentation. It's quite informative. Can I ask about how you are demonstrating whether an organ chip is as good or better than the rat? Are you comparing directly against rat data to look for concordance, or will you take a more holistic approach as was discussed in the validation framework from ICFAM?
Well, obviously I would put some of the validation framework in because I co-chaired that report. It would be have my thinking. We're just starting on that. Okay, we're starting on that. We just had our first meeting last week. Obviously, we're working with tissues, and we do have tissues in our lab already for a couple of years. For example, we use the tissues chip to look at whether the COVID virus can cross the placenta. So we have a lot of experience with it. And we actually, the person that's best, Chung Shi, who chairs the alternative methods working group and is really the brains behind this, not me. But he looked at the chip itself and thinks it will be good for mixtures and food. But beyond that, we're certainly open as we work on this project to include anyone's ideas or not, because really the more people that are included in things that you work on, the better the project. So, but like I said, we just met for the first time last week. We're meeting monthly. BFR is just developing it. We just sent the information to Singapore, and they have other partner there on how much the chip actually, to be honest, is much cheaper than, I shouldn't say this, the emulate chip and stuff. So the whole program is much cheaper than some of the other chips. And the other thing about the tissues chip is that you have linkers in it. So you can link up, they're actually validated linking the gut and the liver, but they can link up to 6 chips. And if you walk into the tissues, lab in Germany, it looks like almost what you order in a restaurant. They've got all the different chips that you can add to their, tissues that you can add to their chips, and you can kind of pick one. So I think people we saw at the World Congress, tissues talked about their liver, thyroid, their skin, liver, thyroid chip, and which was looking for good cosmetics. So they've been presenting that one to our cosmetics group to see if that would be something they'd be interested in. So it's a pretty neat, easier and a little bit more economic chip than some of the others that we have a lot of experience with a lot of the different chips there. So, and a nice company to work with. And plus BFR took over Ilmerac, and they're German, and Tissues is a German company too. So that's it. Yeah.
All right, we have one more.
Okay.
This is from Sherman McFarland. Does CFSAN or FDA still use dogs in their studies? And if so, is there a timeline to stop doing that?
Well, I can't speak for, we have not gotten a dog study in CFSAN since 2018. Okay. We have no, I mean, we have the old red book that I don't think you can find on a website anymore, but it still was never a guidance. It was just some information. So no, we have not gotten dogs. We would not encourage someone to come in for a direct food additive petition, but I'm not speaking for the rest of the agency, and they'd be really mad at me if I tried to answer that question. So.
Okay. Thank you, Susie, for that excellent presentation. Apologies that we lost room video at some point during that. Okay, now I would like to turn it over to Dr. Eka Rufer. She is from the Office of Science Engineering Laboratories at the Center for Devices and Radiological Health Research at the US FDA. And hopefully she can go ahead and share her screen. Let me escape out. Hang on 1 second. Let me just escape out of this and then move this over. The joys of a hybrid meeting. There we go.
I would be there in person, but I'm in California, so it's a bit of a hike.
Well, we are delighted to have you here. Okay, we can see your slides in the room, so you can go ahead, and I will find this and mute myself. So yeah, please go ahead.
Okay, thank you. Thanks very much for asking me to speak today. I'm going to be giving you a little bit of an update on research at CDRH to evaluate medical device safety using alternative approaches. Of course, the standard government disclaimer that what I'm going to be talking about today is a research perspective, an opinion of my own, and it is not meant to be agency policy. In addition, the mention of any commercial projects or specific products or pictures shouldn't be considered endorsements by the agency either. And so within CDRH, or the Center for Devices and Radiological Health, I sit within the Office of Science and Engineering Laboratories, or OSEL. And you can really think of this as the research arm within CDRH, really aiming to accelerate patient access to innovative, safe, and effective medical devices. And we are organized into 20 program areas that range from biocompatibility and toxicology to AI and machine learning to digital pathology. And I, of course, lead the biocompatibility and toxicology program, which is what I'll be talking a little bit about today. And but of course, work very closely with other programs that really support the biocompatibility and toxicological aspects of medical devices from a research perspective, and that's the chemical characterization and sterility infection control teams. And today I'm going to talk to you a little bit about our plans to use regulatory science to modernize biocompatibility evaluations and, importantly, reduce animal use through alternative methods. While much of what I'll talk about today isn't going to be new to the toxicology world as a whole, though they may be new to CDRH, and I think many of them can have significant impacts on reducing animal use and streamlining the biocompatibility and evaluation process. Now, importantly, OCEL's output is not just research for the sake of research. Our output is ultimately meant to be regulatory science tools, or RSTs, that can be applied for medical device development. Now, what are RSTs? RSTs are really peer-reviewed tools that may not be qualified like a medical device development tool or a method within a standard. But they do provide benefit over publications because they really provide instructions on how to apply the tool, which oftentimes aren't present in publications. And this is because we really want these RSTs to be applied, and then we can further develop them rather than them languishing in a journal somewhere. In addition, stakeholder feedback really does play a key role in what RSTs we work on. And our intent is really that eventually these RSTs make their way into the MDDT category or become widely accepted from a regulatory standpoint or are added to standards. And many of these projects are being actively conducted with collaborators, because of course that helps everything move faster, with whether it be academia, healthcare providers, other government agencies, and industry to support these. All of our RSTs are released publicly, and they can be found on FDA's website. I've put a link here. And these range from lab methods to best practices to virtual and physical phantoms, computer modeling, and simulation. However, it is important to note that if you are considering using a tool from this catalog in your marketing submission, note that these tools have not been qualified as medical device development tools, so there may be questions. So we highly recommend you request feedback for submissions as part of a Q submission program. And now I know that not everyone here are working in the medical device arena. So I wanted to talk a little bit from a very high level about how medical device biocompatibility is addressed today. It's really driven through FDA guidance and ISO 10993 Part 1 that are used to determine what endpoints should be evaluated based on a table very similar to this. I've, of course, made some minor modifications. And then the endpoints evaluated for a particular device depend on the nature and duration of body contact for that device, which is, of course, based on risk. And then evaluation of each endpoint for the device is necessary to determine for both new devices as well as changes to existing devices where the change can't otherwise be justified to be safe. Examples of device changes might be a change in a manufacturing process, an alteration to a material formulation or similar, or a change in sterilization process. Importantly, evaluation does not necessarily mean testing, but this is often the default in practice. And then in addition, if you look at this chart here, I've color coded them to try to identify which ones require animal testing and which ones allow for other methods. So currently many endpoints do require animal tests, as shown in the red cells with the red dots on the table. Some of the other endpoints can be evaluated using information on chemistry from the devices, such as extractable and leachable testing on a device extract. And my team is really asking critical questions about the way we assess the safety of medical devices today and looking at alternative methods that could be used to evaluate these endpoints without the use of animals or that can reduce the use of animals. And by asking these questions, the knowledge gap, we can identify the knowledge gaps that then we can determine what research should be done to fill those knowledge gaps so we can really move out of these animal tests. And today, I just want to talk through a very high level of some of the questions we're asking that may be able to help modernize and streamline the approach to evaluate medical device safety testing using alternative methods, potentially in the nearer term. And I know there's a lot of work going on regarding in vitro methods, which is very important, but I'm going to focus my discussion today on some other efforts to address regulatory science gaps using alternative approaches. Now, as I said earlier, these might not be new, but my thinking is that they may be more actionable in the shorter term and have significant benefit in reducing animal use before the other methods are validated, verified, and qualified. One major effort we're looking into is using weight of evidence. Weight of evidence is commonly used in other industries, such as food, environmental evaluations, chemical industries, et cetera. And so we're wondering if there's specific contexts where this line of thinking can be applied. And we're looking at this for several different scenarios. I'll only be discussing one in any detail today. And that is for a specific change in sterilization modality that is expected due to upcoming EPA regulations. And I'll provide more detail on that in upcoming slides. But there have also been several frameworks published for medical devices that we are investigating to determine whether they can be more broadly applied and whether they need to be adapted and how that would be done. And then finally, we're evaluating whether there are other scenarios where we can identify so-called safe harbor materials, where maybe material and chemistry testing can be used to demonstrate a material is equivalent to those which we already have biocompatibility data for, so that this information can be used to support the new device or a change for a specific material. And in addition to that, we're also looking at evaluating how chemistry information from the device may be able to be used to evaluate local toxicity endpoints that currently use animal testing. So getting into the question of how can we use weight of evidence to evaluate medical device safety and to talk about the specific situation with vaporized hydrogen peroxide. So there is an upcoming EPA regulation to limit ethylene oxide emissions. And ethylene oxide is a very important sterilant for the medical device industry. Something like 50% of medical devices that are sterilized with ethylene oxide. So while this change is beneficial to the environment, it can lead to a lot of additional testing. And this tool really aims to provide a technical rationale that can increase confidence of this assessment to determine when testing is and when it isn't, to prevent the default of just do the testing for all of these endpoints I showed in that table earlier. And this VHP risk assessment tool is really a framework for manufacturers and reviewers to ensure consistent testing of legacy devices. So these are devices that are already on the market that can help to assure reasonable safety when they're switching from the sterilization modality, between these two sterilization modality. And the initial tool really focuses on VHP and metal and durable polymers because that's where we've got data on. And it provides a stepwise approach to address biocompatibility risks in a consistent, least burdensome manner. And this approach is really accomplished through weight of evidence. And so this tool really uses multiple lines of evidence to be considered to evaluate whether or not additional testing is needed. The first line of evidence is really, it has a clearly defined scope, is in that it is only meant to be applied to devices that are already marketed. So a brand new device, if they're going to start out with sterilization with VHP, they should just do the standard tests as they are today. And then the other important piece is that it's the only change is from ethylene oxide to vaporized hydrogen peroxide on the materials that we've defined to be within scope, and that's durable polymers and metals. And this is really based on extensive scientific knowledge and data we've been able to collect on how VHP can affect these materials. And that's in addition to some other information that would be gathered on the materials and potential extractables or leachables that come out of the device, and helping to understand whether or not, what is the material's compatibility with vaporized hydrogen peroxide by looking at certain surface characteristics and performance characteristics, looking at potential changes in chemistry or what a patient or a user might be exposed to, and then finally evaluating safety risk through weight of evidence. And finally, controlling residual hydrogen peroxide, because many tissues do not like hydrogen peroxide either. And I'm not going to talk about we've worked this into a process flow, which is shown here on the right, very small with no expectation that anyone is reading it. because I'm not going to be talking about it today, but I encourage you to watch on FDA's website for more information that is to be presented in an upcoming sterilization town hall on this tool in both October and November, where we'll go into much greater depth on this. Okay, and next getting into the question of, can chemistry information on a device be used to evaluate local endpoints? And so to step back for a moment and ask and explain how sensitization and irritation potential of medical devices, how they are evaluated today, is that essentially a medical device is soaked in a solvent, oftentimes saline or sesame oil, and that extract then that's created is used in that animal test, whether it's a rabbit for the intracutaneous reactivity or irritation test, or a guinea pig that then goes through the guinea pig maximization tests. And so in my eyes, because these tests are conducted on a device extract and not on the device itself, there are probably many cases where an analysis of that extract and looking at those the chemicals that are actually present, which is already available oftentimes as part of the biocompatibility evaluation, may be able to be leveraged to make predictions on these endpoints. So I know a lot of other sectors have been doing a lot of work on this, and we don't want to reinvent the wheel. So where have alternative methods been used in other sectors? Well, there are quite a few sectors where a lot of different approaches have been used. We're looking at sectors such as packaging for parental drug products, pesticides, personal care products, and even more recently, consumer electronics have been developing and using alternative approaches over the last several decades. And in my opinion, each of these shows promise for different uses within the medical device realm. And from the irritation perspective, of course, some of these are use in vitro testing, human testing, hazard assessment, as well as there's some threshold limits such as PQRI that have been developed. And it's similar for the sensitization aspect, although additional in silico models, defined approaches, quantitative risk assessment methods and the like have been, I would say, published extensively within these other sectors. And so today I'll talk a little bit more about the risk assessment and threshold aspects and how we might be able to look at those. So the first question that I always hear, which I think is a very important question in order to use chemistry for some of these local endpoints. The question becomes, well, is the chemistry testing that is already conducted on a medical device sensitive enough to support evaluation of these endpoints? Because the analytical evaluation threshold, or essentially the levels of quantification and how sensitive that testing is really based on systemic toxicity and thresholds of toxicological concern. And there's not been a threshold value for medical devices that is currently accepted. So there's some work to do around that. And there has been, as an example, dermal sensitization thresholds that have been published for other sectors, as well as I've talked about the PQRI thresholds. It's unclear how these relate to medical devices. So our intent is to look into this and start to answer that question, so that in situations where it makes sense that we can potentially use this or identify how the chemistry needs to change if that were to be an approach we were a path we were to go down. And the other piece is that I think fits in kind of at the same time is, how can chemistry be used to evaluate skin irritation and sensitization risk if we're feeling comfortable about the sensitivity of the chemistry testing? Is it a threshold value approach? Is it a hazard assessment or some sort of quantitative risk assessment? And it's likely to be different for both of these endpoints that I really don't have time to get into today. But there's a lot of questions that need to be answered that if we're to say go down the quantitative risk assessment path around, what uncertainty factors are appropriate. I mean, a lot of these methods were otherwise evaluated for intact skin exposure. However, in the medical device world, we're looking at a range of devices that might be from, something that touches intact skin to something that's implanted in the body, maybe in the within the skull and is coming into contact with the brain, or you can think of orthopedic implants or cardiac implants. And so there's a lot of questions around uncertainty factors and how exposure would be calculated in order to use that. So my team intends to spend the near future evaluating whether this is something we can use. So in the near term, we'll be looking at numerous case studies to identify an action plan and how we can answer these questions, and if and where these approaches may be applicable. And so you may be asking, I hope you are, how you can contribute. So as I mentioned earlier, stakeholders provide really important feedback on what is needed, where those regulatory science gaps sit, and how it should be prioritized. So I encourage you to reach out to me on feedback you may have. In addition, collaboration can really increase our capacity and develop these alternative methods more quickly. So I encourage you to reach out. And with that, I will take any questions.
Fantastic. Thank you so much, Echo. If you want to go ahead and stop sharing, we can get back to the Zoom video. And I'm aware that we're running a little bit behind schedule, but we do want to make sure that we have ample time for discussion and for questions. So we're going to do questions, and then we're going to take a break, and then we will come back with Warren's presentation. So we'll start with questions in the room. Go ahead.
Great. Thanks, Echo. This is Amy Klippinger from PETA. Good to see you. Just a question. It was interesting to hear about the chemical characterization for the skin irritation endpoint. And I'm wondering if you're able to share if there's been any progress with the in vitro reconstructed human tissue model, which is now accepted in Europe and China and Japan and Australia and so forth.
So some of you may be aware, but Shelby Skoog actually presented some very interesting data on this that at SOT this year, showing that really in those handful of chemicals, it didn't have the greatest predictivity from a positive irritant standpoint. And so we're really at a point where we're looking for additional data. So if anybody has data that they can share, please reach out. Let's chat about that.
Positives data.
Yeah, that's what she showed. And it wasn't, what she showed was, I think it was like a 33% predictivity rate based on some known irritants that were input into the assay and compared to animals. So I encourage you to look at that poster. And if you don't have it, I can share it with you.
I have it. Thanks.
Okay, thank you. The next question comes from online. Are any of the large device companies using NAMs in device safety testing?
I can't speak to policy. That's that's definitely, one thing there. I'm not actually sure. I from my informal screening, there are some I think that are using them for R&D purposes. But I can't give any specific details on what specific companies are doing. And we do, as an agency, I think we just updated our webpage on some of these alternative methods. We are encouraging the use of alternative approaches. However, there are some, not many that are widely available, and they need to be appropriately validated and qualified in order to be accepted.
Okay, thank you. I don't see any other questions online. Do we have any other questions in the room before we take a short break? Nope. Okay. Thank you so much, Echo. Really appreciate your presentation and your contributions. And so everyone to take a 10-minute break to get us a little closer to being back on schedule. So we'll come back here at 2:42 and resume with Warren Casey's presentation. Okay, we are going to go ahead and resume our program. So Dr. Warren Casey is the Director of Strategic Partnerships at the NIHS Division of Translational Toxicology. Also the former NICEDM Director and Executive Director of ICFAM and the current NIEHS rep to ICFAM. And he's going to talk to us about a really exciting program that he's been helping lead.
Thank you very much, Nicole. Thanks to everybody for joining online, and especially for those in the room for this, our 10th anniversary of Public Forum. I realized that when I was looking at my date. I was like, wow. I think it's hard to put in perspective how important this event was back in 2014. We didn't officially reinvent ICFAM until 2016. So agreeing to do this in 2014 was really the first publicly facing effort to say, yeah, we're serious about changing the committee. And it was not well received, and understandably so. ICFAM was not a popular committee back then. It wasn't popular with public. It wasn't popular with a lot of stakeholders. It wasn't popular with Congress. And to be honest, I think Susie can attest to this, it wasn't popular with a lot of the agencies that were on ICFAM. Some of them were trying to get off. Okay, so yeah, between that and the sensitivity around animal testing, a lot could have gone wrong. Having people actually come for a face-to-face meeting on NIH campus facilities. So we tried it and it turned out okay. And I think the thing that has surprised almost everybody, especially people in ICFAM, is how useful this event is for understanding what's going on in the other agencies. I think maybe people assume that we all know what's happening in other agencies, and it's just not the case. This is where we come to find out a lot of times what's happening in other groups. So I'm very happy to be here and happy you guys are here. And I'm going to talk about this project for military exposures and toxin history evaluation in U.S. service members, which is Prometheus. But to give you a little bit of context on how this fits in with DTT and NIHS. So this is the mission statement of our division to improve public health through developing data and knowledge that are translatable, predictive, and timely. And to me, the translatable predictive means human relevant. And that's something that we haven't been, toxicologists in general, haven't been exceptionally great at. We've done the best we can, but we are far from being highly accurate in the ability to predict how a chemical is going to interact with the human body. And it has to be impactful. We have to do research and deliver the results in a timely manner so that they can actually make a difference. As Allison was mentioning, if we do, it takes on average about 12 years to get a 2-year bioassay out and reported. And even then, we sometimes don't have a very clear picture of what's going on. We do 2 species, 2 sexes, and a lot of times it's not a uniform response. We still don't know. So we need to be able in the future to actually use human relevant approaches to predict human toxicity. So that's what mission statement is. We have a framework where we have 4 research areas, capabilities, responsive research, exposure, and then health effects innovations that focus on specific toxicities or disease. And each one of these has a program management team or 2, or so this one has 2 program management teams, which focus on work in those particular areas. This project that I'm going to talk about could be a lot of different places, but it is located here in the Carcinogenicity Health Effects Innovation Program team. And this is the translational toxicology pipeline. You may see this a couple more times throughout this event. The thing that was missing a few years ago from this diagram was, my point. Is there a pointer on here? Was, oh, there it is. It's not showing up on the screen. Ah, okay.
Right.
Okay, so was this icon here. So several years ago, this was exactly the same, but we did not have the human studies. And we thought it was really important to put it on there because something that occurred to me once I started this project was that we use surprisingly little human data. Like that's not what toxicologists do. We work with animal data, and that's kind of a chicken egg thing. We work with animal data because that's what we have. That's what we become used to. So we don't really go out of our way to incorporate human data. And the other thing is that there's not a lot of human data out there to incorporate. So I think in the future, without a doubt, human-based toxicology is going to be the future of toxicology. However many years in the future, there will come a time when we understand human physiology to the point we understand the etiology and pathophysiology of toxicity and disease so well that we can generate computational models, in vitro models, and trust the results from those much better than we can the results from an animal test. I'm not even close to there yet, but that's where we'll eventually go. And to get there, we have to start using human data and be comfortable using human data as toxicologists. So that's the challenge. I've given lots of talks where we talk about the challenges of changing things, and comparing results to animal tests is always a problem, as is the availability of human data. People often respond, well, you have ER information on overdoses, or you have pharma clinical trials. Neither one of those are viable sources of the kind of data that we need. So that's where this particular project comes in. This Prometheus project is run by the department. It was founded by Dr. Craig Shriver, who's the director of the Martha Cancer Institute right across the street, Walter Reed. He also started another project I'll talk about called Apollo. But he started this in response to a challenge from the White House and Congress also, to really bring the resources of the federal government to bear on this question of how do environmental exposures affect service members and veterans. Of course, that's applicable to everybody else as well. And we're really trying to develop actionable tools that can be used to monitor people's health, monitor exposures, and let them know when they're getting close to an increased risk of a certain type of disease. So those are kind of the high-level goals. This is part of the 2022 Cancer Moonshot Initiative, where it talks about understanding and preventing toxic environmental exposures. There's $47 million set aside for FY24. Depending on how you look at it, $47 million is not a lot of money for this size of a project. I'm not saying it's not significant, but the Department of Defense is putting a lot more than $47 million into this project, as are other agencies which are contributing in kind and directly. The VA is putting a ton of effort into other resources that are being utilized in this program. So the federal agencies' expenditures for this program and related programs is just absolutely enormous, and enormous by DoD standards is enormous. So this is also responsive to the Sergeant First Class Heath Robertson honoring our promise to address Comprehensive Toxics Act of 2022, or PACT Act. SFC Robinson was a medic who 10 years after being deployed in combat area came down with a very rare autoimmune disease, as well as stage 4 adenocarcinoma of the lung. Both were attributed to exposures he experienced while on duty. And that was kind of the impetus for passing this bill, which is really aimed at the VA, but there's a lot of language in there that says we need to do a better job of understanding how exposure is affecting service members and vets. And this is really an umbrella effort. Again, it's funded by the DA, but very much dependent on other agencies, especially VA and NCI. NIEHS is also contributing, and public-private partnerships. So this isn't one program. There are over 60 funded programs, right? 60 funded projects. There are 30 that were funded 2 years ago. The other 30-ish are going to be starting this summer. So I'm going to talk about a few of those that I'm involved. But first, I want to talk about some of those other resources that are available, because it's not just the Prometheus project that will provide a lot of human relevant data. There are a lot of other resources that are being used that are valuable on their own. And probably the most widely known is the Department of Defense Serum Repository. So this has over 70 million samples from over 10 million service members. And it was started in 1986 in response to the AIDS epidemic. They wanted service members to be regularly tested for AIDS in case they needed to give blood on the battlefield under emergency circumstances. So every 2 years, anybody on active duty has their blood taken. So starting in 1986, if you were in the military, you got it when you came into the military, and you have it every 2 years, period. And it's stored in a physical facility, a big warehouse. So my samples are in there. You can actually, and we've tried this, you can actually look up your samples and go to them exactly. So it's highly organized. It's tied to electronic health records. It's tied to a lot of other pieces of information, so you can cross-reference and identify the samples that you actually want to use. And then in 1996, they also implemented a law that the DoD has to test people before and after deployments. So I think it's pretty easy to get the concept of, if you have this longitudinal serum resource every 2 years, if you can understand what people were exposed to, and you know what the outcomes were, you can actually work backwards. You can find all the people with a certain type of cancer that were in a certain area and go back through the serum and incorporate other kind of epidemiological data to get a better understanding of what could have contributed to that disease. The VA also has two resources that help estimate what those exposures were. One's called the Individual Longitudinal Exposure Record, the ILR. All the information there is what's included. This is more of a high level. So this is a VA assigning exposures to a person's record. So this person was in Southwest Asia. Southwest Asia had a high PM 2.5 level for this period of time. So it's more of a VA taking known exposures and associating them with records. The VMOT is the opposite. It's actually being filled out by some online resource, self-reported from veterans, and they get into a lot of more detail about what they were actually exposed to, where they actually were during certain periods. So between the two of these, you have a really good idea of what they could have been exposed to. There's this study that DoD and VA jointly did, military biomarkers research study. And it's an amazing piece of work that hardly gets referenced, unfortunately. So what they did was they first looked at the samples in the DoDSR, determined how far they could go back. Can you take a 20-year-old sample and still do microRNA? Can you still get metabolomics? How long do these things stay stable? Because they're stored at -30, which is not ideal. I mean, obviously you'd like them a lot colder than that, but certain things remain very stable. And so that's the first thing they did. Then they looked at the association between certain exposures, certain known exposures, measured exposures, and biomarkers. And then they looked at the relationship between those biomarkers and health outcomes. And I thought this was really interesting. At phase 4, they started using in vitro biomarkers. They were conducting in vitro studies where they could add the chemicals and see the same biomarkers they saw in the serum. So you can imagine if you could reverse that, you can start conducting this exposure research and project what you should be looking for in the actual serum samples and understand what exposure led to that biomarker. All of this was published in 2 special supplemental journals, Journal of Occupational and Environmental Medicine. There are about 8 or 10 papers in each of those, and it's just fascinating work. Framingham was named after the heart study, but it's a longitudinal research study that's looking for early biomarkers of detection. So what they're doing is they take people with a specifically known type of cancer from the health records. They go back and look at that person's serum at the point that was most proximal to their diagnosis, and then the 2 previous points. And then they also look at the serum after treatment, so after they're declared disease-free. And they use targeted and non-targeted proteomics. The thing that really excites me about this work, those are the cancers they've done so far. This is ongoing. The thing that I think is really exciting is that they only did these two analyses, and it took like 50 microliters. You get a 0.5 ml from the DODSR. So all these samples are still sitting there, already consented, ready to go. So anytime you can find samples that have this kind of history and they're already consented and you can go back into them, that's really valuable. So we're now looking to make sure that we don't actually have to reconsent. But if that's the case, this is going to be, another gold mine opportunity. This is another huge one. This is DoD, VA, and NCI. Again, initiated by Dr. Schreiber. This is the Applied Proteomics Organization Learning and Outcome Network. He likes to come up with these acronyms that don't really roll off the tongue. How did you come up with that? But anyway, Apollo. So what they've done here is anybody that's diagnosed, excuse me, anybody that's diagnosed with cancer in a VA or DoD facility, meaning service members, vets, or their families, this started in 2016. Anybody since then that is diagnosed, they're offered the opportunity to enroll in this program. And what they do is they take the tumor and somatic cells. They do 2 types of whole genome sequencing. They do proteomics on the tumor and in the blood. And so there's enormous amount of information associated with these patients. They don't have any information on the serum. And that's where the next project comes in. So the project I'm going to describe in a few minutes is this Apollo. We're using patients from Apollo 5, which is a pan-cancer study, and taking their serum and going backwards in time to see if we can link exposures measured in serum with disease, with genotype, because we have all that information. Pulling all that data together and figuring out how to analyze it is one of the most challenging aspects of the study. So these are some research programs that are underway now or going to be underway. So the first one is what I just spoke about. This was probably the flagship program of Prometheus. So this pan-cancer, it is focused on deployment-related exposures, meaning that, so the White House has a very high level of interest in understanding exposures related to deployments, especially burn pits. So that's what really drove a lot of this. And the way this is working, we're getting 6 samples from the DoDSR, 1 from when the people were first enrolled in the military, before and after 2 deployments, and then 1 most proximal to being diagnosed. And what I didn't include on here, unfortunately, is we have another sample. So those are 6 are from the DoDSR. We have another 50 ml sample from when they are enrolled. So after they're diagnosed, before they're treated. And then we'll have, so the program is designed to analyze those 7 samples. There will be additional samples available because as they go through this program, blood's collected like every year or after every major step in treatment. So we're using the ILR and we're using a modified version of the VMOTE. The VMOTE is a self-administered online tool. What we've done is we've developed a bespoke version of it with questions specifically to address the questions we want to answer. And this will be administered person to person. They've done this in the past and they have so much better success when somebody's sitting down with somebody. If you have a trained staff member that's administering the questions, you can answer questions about how to interpret something, or you can be triggered to ask additional questions. So that in itself is a significant investment, considering there could be 5,000 people enrolled in this program. So to put the other programs in context, the biomarker research study was very early on, but didn't look at any other serum. Apollo just looks at the tumors, didn't look at any serum. And then the Framingham just looked at serum, but not specifically tumors. And so what we're trying to do with this one is tie all of that together. Doing targeted metabolomics, non-targeted metabolomics, and targeted proteomics. Tying all that with the whole genome sequencing from the tumors and somatic cells. This is a massive, massive effort. Lots of people involved, lots of agencies involved, lots of NGOs, private companies, Blood Pack, HESI, University of Chicago. There are a lot of players in this game, and just coordinating everybody has been a significant challenge in itself. So another one that has been funded and will start this summer stems from this paper by Kangman Zhu is the senior author. He's my collaborator on the next two projects. He does a lot. If you see a paper about cancer statistics in the military, he probably either the first author or the last author. That's basically what he does, is analyze cancer statistics for military and VA. So they did this study where they used DODSR samples and looked at people who had colorectal cancer. And the idea was to identify early biomarkers. What they didn't say, which I was surprised, was they didn't really dig in on the fact that the average age of the patients was 40 years old. That's early onset colorectal cancer. That's the definition of early onset. And it turns out the military is a great resource for looking at early onset diseases. There's about 1.3 million active duty service members now. And the average age is 38 and a half. So you have such a skewed age distribution that when people get diagnosed with disease, they're probably going to be young. So this study looked at the most proximal sample and then the two previous, as well as an initial. And again, those samples are still sitting there. So I got funding to do some additional analysis. So we're going to do microRNA profiling that's being conducted at the American Genome Center, which is the DoD's one of their big sequencing facilities. They run like 1,000, 1,500 whole genomes a week. They do a lot of work. So they're going to do the microRNA profiling. And then we're doing the exposome using regular mass spec, but also ion mobility mass spec, which is supposed to give a better separation. It's not my field, but supposed to give a much better separation of the different molecules. This is with Aaron Baker and Yvonne Rusin. You have no idea how difficult it was for me to put that UNC logo in my slide. Because I'm an NC State fan. Okay, so the next one that hasn't been approved yet, but is actually the one that started a lot of this. And then the first one I talked about became the major project. So we've gone back to this one to try to finish it up. And this is the looking at early onset breast cancer. Early onset breast cancer is the incidence is increasing, is disproportionately affects African Americans and Hispanic women. It is much more aggressive. People tend to be at a higher stage when they're diagnosed, probably because they're young, they're not expecting it. I think the mammography screenings have the suggested age has been lowered to 40 now because of this increase in early onset breast cancer. You tend to have higher incidence of triple negatives that are hard to treat, triple negatives and HER2 overexpressed. So this is it's a significant issue for everybody, but especially for the military. The average age, I said it on the other slide, but the average age for somebody on active duty being diagnosed with breast cancer is 40 years old. That's actually like 39 something, which is pretty significant. It has a significant impact on readiness. It has a significant impact on their ability to keep performing their job. So if we can figure out ways to identify early biomarkers or some kind of precondition that would allow change in job duties, if it's associated with an environmental exposure, if we could figure out a way to, okay, I'm getting okay, here we go. Anyway, so we're doing, what makes this interesting is it's deployment agnostic. It doesn't matter. We're going to find out where they deploy, but that's not the basis of the selection. And the other thing that makes it perhaps challenging, we're asking for all samples. DODSR has a policy, you only get 4. We've already got an exception to get 6. Now I'm going to ask for 12. I don't think it's ever been done before, but we'll see. We're also doing prospective studies using a blood collection device. One draw, you stick it on your arm, push a button, you get 150 microliters of blood on filter paper. And these are the 2 scenarios that we're doing right now. One's naval flight line exposures on an aircraft carrier. Okay, I'm going. Nicole's looking very nervous over there. Okay. Okay, and longitudinal monitoring of people being, when they come into the military at Fort Liberty with the 82nd Airborne, they have lots of deployments. So we're going to keep tracking them on their deployments. This is one that we're no longer involved with. Terre Vincent Hall in the VA and Sioux Fenton used to be with DTT. This is one of their projects. And I'll admit, the only reason I did this is to get the NC State vote. Okay, I'll wrap it up quick. Lots of people to thank on this. Jerry Lee was instrumental. He's the one that connected me with Dr. Shriver and essentially vouched for me. Otherwise, it's really difficult to get access to these resources. Dr. Shriver has been amazing. I don't know. He's a surgical oncologist, a practicing surgical oncologist, heads up the cancer center, also heads up Apollo in Framingham. I don't know how he has the bandwidth. This is the CARCHI team, and this is the last picture of the Prometheus group at our retreat. And now I'm finished.
All right, thank you, Warren. That was excellent. One question from Connie King at FDA. Very exciting project in collaboration with DoD. Curious as how forthcoming DoD is providing all information regarding potential exposure. Is this exclusively for exposure during deployment only, or what about exposure related to contaminations at military bases, thinking Fort Liberty? Yes.
So I think they're being very forthcoming. I think there's a lot of information that's missing. I think maybe at certain times they may have not have been so forthcoming, but what we're realizing now is that exposures that happen in garrison can be just as bad, if not worse, than what happens when you're deployed to PFAS, jet fuel. There's lots of stuff that happens in garrison during training. So I've been amazed at how willing they are to let us have the samples and let us go to Fort Liberty and get on an aircraft carrier. So I've been pleasantly surprised at the willingness of everybody to work together to help solve this.
Great. Thank you so much, Warren. Okay, now I'm going to turn it over to Will Gwynn. Dr. Will Gwynn is also from the Division of Translational Toxicology at NIEHS. And hang on, I just need to go back to the Zoom. Shoot. Share screen first. Okay, so turning it over to Dr. Gwynn to take us through some other excellent work going on at DTT.
And the pointer. Thanks, Nicole, and thanks for having me. So I'm in the systems tox branch of DTT, and I'm going to give you a taste of some of the work we're doing with in vitro lung models, specifically ALI approaches. Is this the oh. Sorry. So I know Warren already went through this a bit. So again, in terms of our structure, we have 4 what we call strategic areas of focus shown here in the 4 boxes. And within those 4 areas of focus, we have 10 programs. And you can see over here, top left, we have 3 programs that are under the exposure-based research programs. And one of those programs is the Occupational and Inhalation Exposures, or OIE program, which I am a member of. And the work and the studies I'm going to describe are all fall under and are managed by the OIE program. So here is the OIE program management team, or PMT. So there's 7 of us now, actually. Rebecca was one of our postdocs, and she recently left to go to FDA. But we have diversity in terms of, we have toxicologists, we have pathologists, we have vets, and we have those in program operations within DTT, including cores. Here are some of our many program partners and stakeholders you can see. One of the big ones, important ones for this work that I'm going to show is Battelle, which is one of our contract labs. So here we have the driving problem statement for the program. So hazard characterization of inhalation exposures is critical to creating a safe living, working environment and reducing disease burden. And within the program, we have 3 main objectives. So the in vitro ALI work fall under objective number 2, which is to expand our capabilities for predicting adverse health effects. So the program is currently evaluating novel or alternative technologies to investigate human relevant inhalation toxicity. These include in vitro airway models like ALI, as well as some of the lung microphysiological systems, such as some of the lung on a chip systems. But what we've been mostly focusing on, what I'm going to talk about today, is the ALI work that's ongoing. And so in this middle panel here, so standard ALI cultures consist of differentiated airway epithelial cells, which are grown on a microporous membrane. And the basal side is bathed in culture media, whereas the apical surface is exposed to air. And you can see in this picture here, you have your ciliated epithelial cells, you have goblet cells, so these tissues can produce mucus. You also have your basal cells. And these cultures are typically derived from a primary donor, tracheobronchial, bronchial, or alveolar cells. Multiple donors are available. They're also, you can also test rodent cells. And so you can get these commercially. So there's many vendors that sell these tissues, or many people make them in-house in their lab. And so there are sophisticated exposure systems, many of which are commercially available, that can be used to expose these cultures to vapors, gases, aerosols, particles. You can really have the physiologically relevant exposure route. Depending on the exposure system that you use, you can increase the throughput in terms of the number of wells or samples or doses that you can expose simultaneously. So what we decided to do was to do what we're calling a pilot or proof of concept study testing 2,3-pentane-dione with the ALI models. And so a little bit of background. So 2,3-pentane-dione, which I'm going to call PD moving forward, is a major and highly volatile component of artificial butter flavorings. So it's been used as a replacement for 2,3-butane-dione, which is more commonly known as diacetyl, which is known to cause or has been known to cause a form of potentially lethal airway fibrosis called obliterative bronchiolitis, also called bronchiolitis obliterans, in exposed workers, especially in the microwave popcorn industry. It's also had the name popcorn lung. So PD and diacetyl are structurally similar. So they're both alpha diketones and they exhibit similar inhalation toxicity in vivo. So I've been working with these chemicals for many years now, and we've shown that inhalation exposure causes airway toxicity in rodents, rats, and mice, including adverse bronchial and bronchiolar effects. And in rats, we can induce OB-like fibrotic lesions that recapitulate the lesions that you see in humans. And these are just the structures here on the right. And then there are in vitro human airway tox data for diacetyl, which we've used to help elucidate some of the mechanisms driving airway injury and fibrosis. These were mostly done using tracheobronchial epithelial-derived cultures. But we have very limited data for 2,3-pentane-dione. But with that said, we would expect similar in vitro tox effects based on what we know with diacetyl. So our project-specific objectives were really twofold. One was to characterize or optimize a VitraCell 48 2.0 plus exposure system. So we wanted to do method development, validation, do the initial testing of the exposure system and vapor generation with the test article 2,3-pentane-dione. And then next we exposed human and rat ALI cultures, which were tracheobronchial epithelial cell derived, to look at airway toxicity in vitro, as well as between species. And really the idea, we want to, ideally we'd like to be able to extrapolate between what we see rat in vivo to rat in vitro to human in vitro. And so this study was really divided into two phases. One was feasibility study. So we did a lot of troubleshooting of technical challenges that we encountered and optimization. And then the next phase is what we're calling a definitive study, where we had a lot of study refinement based on many lessons learned in the feasibility study. And so the rationale for test article selection is that PD has been well characterized in vivo and is relatively straightforward to work with from a chemistry perspective in terms of being able to generate stable vapor atmospheres within the exposure system. And we also know this from all the in vivo work that we've done in the past with 2,3-pentane-dione. And then also there's currently, as I mentioned, very little human in vitro ALI data for 2,3-pentane-dione, but there have been multiple studies conducted with diacetyl. So really the idea is that we could use that data as a guide for study design and anticipated findings, since the two are so chemically similar. So this is the VitraCell 48 2.0 plus exposure system. that we're using. You can see it set up to the right here. Doesn't seem like why is the arrow not? There we go. Okay, so you can see it set up in this photo on the right at Battelle, again, our contract lab. And on the left is the vapor generation system, and then the exposure module on the right, which is also shown here at the bottom. And so basically what we have is you have the vapor generation here. The vapor is then directed into the exposure module. And we have GC monitoring online and real time of the exposure concentrations. And so this exposure systems allows you to expose up to 56 wells at once. And so in the case of this study, we're able to do 8 concentrations and 7 replicates per concentration. And one of those is the air control. So actually 7 test article concentrations. And then there's a dilution manifold that can be used to aid in dose spacing when you're selecting your concentrations. So this is an outline of the definitive study. So we used human and rat ALI airway cultures from MadTech, so vendor called Epi Airway. We used a single donor and a Wistar strain for rat, and these were all normal tracheobronchial epithelial cell derived cultures. We then exposed for 6 hours to 7 concentrations and an air control. And so the exposure duration, the exposure concentrations, as well as the rat strain were all based on the in vivo data and the in vivo studies that we had done in the past in rodents. And typically ALI exposures are, an hour or less. So another thing we wanted to push kind of test the system, to push the system to see, and we actually conducted earlier on some preliminary studies where we were able to find that exposure for that length of time, 6 hours to just the air control, had no adverse impacts on the tissues. One of the things that we learned in the feasibility study is that exposure concentrations above 240 ppm were just, we saw excessive cytotoxicity and tissue destruction. And so those were removed for the definitive study. So we had 2 collection time points. One was immediately after exposure, and then one was about 18 hours. We collected apical wash, as well as the basolateral culture media samples, collected tissues, and we also collected tissue lysates. And so here are the endpoints that we were looking at. So we did chemistry, so we looked at external concentrations of PD in the apical and media samples. We looked at the test article in the tissue lysates using GCMS. We used TIR and LDH and adenylate kinase assays to look at airway cytotoxicity, cell viability, indicators of injury. We did histology, H&E, and periodic acid shift, the stain for mucins, to look at airway injury histopathologically. We use multiplex assays to look at secreted biomarkers, and we also did some immunohistochemistry. So I'm not going to show you some of this data. We're still is not complete. So I'm only going to show you a little bit of the data that we have so far. And importantly, the toxicity endpoints that were selected are all relevant to in vivo rat, both for diacetyl and PD, as well as in vitro human airway findings for diacetyl and a AOP. And the AOP is AOP 280, which is alpha diketone induced bronchiolitis obliterans. And so one thing we wanted to do was try to use, we used toxicity endpoints to try to match or model key events in the in this AOP. So for example, this key event, the second key event, airway epithelial injury, we can use TEER, we can do cytotoxicity, cell viability assays, as well as histopathology. For the next key event for inflammation, we're doing a multiplex assay looking at various secreted biomarkers, cytokines, chemokines, and so forth. An important thing to keep in mind, though, and this are some limitations of standard, what I'm calling standard ALI models, ALI airway cultures, is that there are no recruited inflammatory immune cells in the system. So really you're relying on cytokines, chemokines, secreted proteins as an indicator of inflammation or pro-inflammatory response. And then also, if you look at the last key event, it's very challenging to model, aberrant repair, remodeling, and fibrosis using standard ALI cultures. There are some models that incorporate fibroblasts and some stromal matrix components, but it's still very difficult to achieve the remodeling that you see with fibrosis. And there are, currently, for example, for those of you familiar with the human cut, precision cut lung slices, so it's interesting. So those tissues include immune cells and have been shown to be able to mimic, so you can actually induce fibrosis in the precision cut lung slices after an ALI exposure using like a Mason trichrome staining for collagen. So this table is just showing, this is the exposure data, and this is just to show you that you can see that for all the target concentrations, we're able to achieve almost 100% of our target with a limit of detection of about 2 ppm. So this is showing our TEER data, so transepithelial electrical resistance. So TEER is a measurement of barrier function and permeability. So it's an indicator of tissue integrity, viability. And so you can see on the left here is human. So we really see a threshold. So we see decreased here at both time points at concentrations of 130 ppm and above. And then on the right, rat, we actually see decreased TEER at 70 and above, but it's interesting because for some of the concentrations, you can see we do get a little bit of recovery at the second time point. So this is showing you some of the histopath images. This is human. And so what we see, and this is 0 hours, so this is immediately after exposure. And so you'll see at concentrations of 130 ppm and above, we see degenerative effects. So we see degeneration, necrosis, as well as loss of cilia. And then we start to see some denudation of the tissues at the highest concentration. You can see in the box on what's boxed up there, you can see that's to show you the cilia on the surface. But I want to emphasize this is a single donor. So this is a male Caucasian. This is one of the MatTek cultures, MatTek donors. And so, this is a discussion we had is, how much, you would want to repeat this in multiple donors to gain confidence. And, what is the minimum number of donors to, have confidence in results. And I know I've had discussions with Sean about this and so forth. And then this is rat. Again, immediately after exposures, and like in humans, at 130 and higher, we start to see degeneration, necrosis, as well as loss of cilia. It was interesting in that the effects were more severe, or the damage was more severe in the rats than in the humans. I'm not showing, for the sake of time, I'm not showing the 18-hour time points for either. But something we need to consider moving forward is that the age of the rat cultures were they were younger, less mature, and less differentiated than we expected. And this is a image of one of the controls, and you can see there's a little bit of cilia on the apical surface, but not as much as we had expected. And so we found out that actually the optimal age is about 3 weeks instead of 2 weeks for the rat culture. So that's something we're considering moving forward. So just a results summary. I already showed you, so we see decreased here. We see increased histopath effects. And importantly, these type of effects are similar to what we see in vivo in the rodents, these sorts of effects. I didn't show the data, but we also see increased cytotoxicity based on measurements of LDH and adenylate kinase release. And then I didn't show any of the chemistry, but we see increased test article in the basolateral culture media samples at 0 hours, so immediately after exposure with increasing exposure concentration, but we see very low levels in the apical samples and lysates. But our theoretical max deposition in the culture media is about 12 to 27% for human and 10 to 15% for rat. So for example, in rat, we have a presumably 90% that's being taken up by the cells, by the cultures. And so a question is, the fate of the pentane dione within the cells. And we're trying, it's been very challenging, but we're trying to measure and look at, is it metabolites that are forming. We've tried looking at one of the major metabolites for 2,3-pentane dione, but that's still ongoing. So additional data that's in progress. We're still waiting on the secreted biomarker data, and then both PAS staining for histopath, and then the immunohistochemistry. So here are our conclusions. So exposure of human and rat cultures to the pentane dione induced concentration-dependent changes in tox parameters that are relevant to what we know happens in vivo for both diacetyl and pentane dione, as well as in vitro in the case of diacetyl, and then some of the key events in the AOP. And so, as I mentioned earlier, it's modeling inflammatory immune cell recruitment and fibrosis using these approaches. It's very challenging, but with that said, we know that airway epithelial injury and the events around that are important initiators of fibrosis. So the idea is to try to capture some of the earlier events before you actually get the true fibrotic response. And then, based on the results of this study, with this exposure system and ALI models, the idea is to have the potential to be used to investigate human relevant inhalation tox in vitro with, multiple different applications as shown here, potential applications. And then we're in the process of doing some further optimization of the human and rat ALI models. Again, I mentioned the age of the cultures, especially in the case of rat, looking at different donors as well as different strains. You can use different cell types. Again, these were tracheobronchial, but you you can change the cell type. And then we also want to, we're doing some studies to develop methods to be able to do transcriptomics. For example, the S1500+ platform that we use extensively in vivo at DTT. We're hoping to apply that to ALI as well. And then just to finish up, for those of you who may or may not have seen it, we recently released a request for information on the use of in vitro lung models in inhalation tox. You can find the federal registry version, or if you go to this link. And so we are asking that all feedback and comments be provided by June 7th. And really the idea is we want to get feedback in terms of how we can prioritize and what we should be focusing more on as we move forward with this work. And there's also talk of potential workshop in this year or next. And then my last point, something else that we're considering is doing a interlab reproducibility study. And we've had discussions with Nicole and NICEDM. So the idea that could we do a study where NICEDM coordinates, testing with multiple ICFAM labs, the 2,3-pentane using their ALI airway models and exposure systems. And really the idea would be to compare and contrast the output tox data, for multiple tox endpoints from different labs. So with that said, just thank you to all my DTT colleagues, as well as all my Battelle colleagues. Thank you all.
Question in the room? Does not look like we have any questions online, but I think we have one in the room.
Great talk, Will. For the rodent cells, was it just a Wistar strain alone, or are there?
Yes, we've only done Wistar because that's the only commercially available one, or the one that MatTech has right now. So right now we're limited in terms of our capabilities, that we're relying on the vendor, so MatTech specifically. And right now Wistar, for what I understand, is the only strain available, but that's something on my list. I need to reach out to them and see if we can get it in other strains potentially.
And while it sounds like the major adverse outcomes in humans from this exposure is bronchiolitis obliterans, do you see, are there known effects of 2,3-pentane or butane in nasal or small airways or the pulmonary region that would also be worth considering as other sources of outcome?
So the bronchiolar compartment would be most relevant, especially for human. In rats, we see bronchial and bronchiolar fibrosis. So you do get fibrosis in more of the upper bronchioles. Nasal, I'm trying to think back to nasal. I don't think much nasal, but we do see some alveolar fibrosis in vivo. In some of the, especially if you expose for 2 weeks and then hold for for a recovery period, you start to see some alveolar fibrosis as well, along with the bronchiolar fibrosis.
Have you guys done any thinking about realizing that it multiplies the cost of the study, but looking in representative models of these other regions of the respiratory tract to see if your dose response or concentration response relationship in tracheobronchial versus bronchiolar versus pulmonary regions is similar, and how that might inform the utility for any predictive model?
Like to. I mean, right now, again, we're reliant.
I realize that's not
It'd be nice
simple
to be
thing
able
to do.
Tailor to the cell type we want.
Yeah,
it's the most easily, the tracheobronchial derived cells are the most easily acquired from the vendors. we've used the full thickness model, epi airway with diacetyl, which includes the fibroblast and matrix. And we get some interesting effects going on there, which we published in 2017. But yeah, I mean, that's something I want to do, is to be able to really dive into the different departments and
Yeah.
how it changes, you know, in terms of the dose response and so forth.
And one last quick question, since you mentioned it. So in the results that you saw in the diacetyl study from 2017, were there any results there, since that was a full thickness model, that, or are there any corresponding results that you would have expected to see but didn't see in the pentane-dione study, where you used an epithelial only model?
Yeah, so one of the interesting findings we saw with diacetyl with the full thickness was what was termed suprabasal basal spongiosis. So you could see it clear, the basal layer within the tissue appeared to be clearly targeted, and you had that distinction between that layer and then the underlying matrix. And so that effect has not been as obvious with the pentane-dione studies, which is not using the full thickness model. And it's also a different donor. So that it was a different donor as well for the diacetyl study. So there's that, and we're still waiting on the multiplex data. So the cytokine, like we saw certain cytokines that were only upregulated or increased when the matrix was there. But if we compared it to the matched donor without the, you know, the standard epi airway without the matrix, we saw differences in which cytokines were increased. So that's another thing we saw. That'll be interesting to see once we get all the cytokine data back.
Absolutely.
So
thanks.
and
Great
I'm
study.
sure we'll chat some more.
Look forward to it.
All right. Okay.
Kind of already answered.
Oh, yeah. Okay.
Basic around the nasal region questions.
But Annie Jarabek had a follow-up from that. So Annie Jarabek just provided an additional comment that physicochemical properties will determine the region, ETTB or PU, of highest interest. So therefore, all three should be evaluated. Just her follow-up comment. And then from Matthew Grog at Air Force, we do a fair amount of epi-alveolar and epi-airway at AFRL. When it comes to tier, we see high variability. What are your thoughts on how many donors and how many replicates should be included in future studies of one or more exposures for publication quality work or regulatory application? It adds up quickly if you're thinking about doing multiple donors.
Yes, that's a great question. And I know Sean has his opinions on tier and, you know, how many donors, and yeah, that's something we don't, you know, how many donors. I think it also depends on which endpoint you're looking at in terms of how many donors you need for most confidence. So I don't think we have a definitive answer to that. I don't know, Sean, did you want to add to that?
That's a discussion that's probably longer than we have here. So I missed the name of whoever that was, but feel free to reach out to me. Happy, or we can discuss it later.
Yeah. And another thing is, you know, some of the, like, for example, I think, and I could be wrong, but I think like the epi-alveolar, I'm not sure if they have as many donors available for that model compared to the more upper airway regions. Yeah, it's still one. I knew it was one before, so it's still one. So that's another challenge, depending on which model you're using, is what's available, unless you, again, make them in the lab in-house.
Great. Thank you so much, Will. Okay, so our next set of presentations is from our colleagues at the National Institute of Standards and Technology, or NIST. And I'm going to invite Elijah Peterson up to the podium to talk to us, to give us a overview of NAMS activities at NIST, and then, okay, we should be good. That's your first slide?
Yep.
Okay.
Just jumping right in. Yeah. I'm happy to be able to share about some work that I'm not doing. So if you have tough questions, I'll put you in touch with the people who are doing it. And it's also very exciting that Darwin and Mandy are here, so my colleagues from a different laboratory in this to talk about some of their MPS work. So they'll be following right along from me. So I'm eager to have them share that with you all today too. So first, I'll talk about something that's come up in some different ICFAM discussions I've been involved in, which is cell counting. Seems kind of obvious, maybe every human cytometer has been done forever, but it's actually a lot of nuances and very challenging, which I'll get into a little bit. Oh, yeah, don't have to find the pointers right here. That's great. Thank you, Will. So this can be relevant for many different applications. In particular here, it's relevant for bioassays. And if you do have questions, Simona Sarkar is the contact person. She's been leading this for many years now. I don't know if it's been a decade, but it's been many years for what is a very common biological measurement. Okay, so here's what you'd love to have is this, where you have good accuracy and precision. That's a bit hard because we don't have appropriate reference material. So we can hope for then is you have precision with unclear accuracy. But what you don't want to have is neither accuracy nor precision. So how do we do this? Because without reference materials, that's, it's kind of in a ground truth, it's kind of challenging. Well, what you could do is to say, well, how does the instrument work relative to itself? So if we do dilution series, how consistent is it across the dilution series and giving us what we want? So this is just one example showing an automated or manual approach. And there are also many different cell viability assays based on different assumptions. And, what is a cell viable? We started getting in a philosophy class there and, some other things. So it's, there's a lot of different ways that this is defined. And, for example, here, this is some control charting data. Things are looking good. Uh-oh, what's happening here? Something's out of range. It's important to have good metrics for making sure things are working well, even in, consistency internal to whatever your approach is. So to do that, there's an ISO standard that came out in 2019 where they describe, okay, here's a process where you can do your dilution series and look at the consistency among these different steps. And then you look at this thing called proportionality index, where you well, if you dilute it by a factor of 10, how is it a tenth of as much, and so forth. So this helps you identify your linear range and how well your approach is working. And again, there's a lot of, I'm just skimming the surface here, lots of nuances in the details and publications. Oops. Why is it not going ahead? I'm clicking, I'll just click here. Okay. So to give kind of bring it back to some of the NAMS world a little bit, this is an example of 3 different approaches. I think my colleagues at NIST did. You'll see, different, the proportionality index is looking pretty good for these 2, a little bit worse for those 2. What's really interesting here is the bias, where even though these 2 methods are pretty good within themselves, they're about 50% different from each other. So from that, you could expect if you're doing some interlab study, people are using different techniques. It wouldn't be surprising if they were varying 50% or more in terms of number of cells. Now, what does that matter in terms of your assay? That's something that's probably important to have a good handle on, because otherwise, this is something we could reasonably expect to vary quite a bit between laboratories, at least with how much you're adding initially. Maybe, they're growing and, getting confluent. It's not so much different at the end, but this is, kind of something important to keep in mind, that this could be impacting your results. And, there's this proportionality index. There's a bunch of hard math behind it. So we try to make it easy for people to adopt. So there's our Shiny app where you can put your data in if you do it based on the format that's suggested, and you'll get information about how well it's looking according to different indicators and recording requirements, similar to what you'd find in the ISO guidance document. So we try to make it easy for everyone to apply this. And again, there's some more papers here that go into a lot more nuances. Another common measurement for different NAMs is flow cytometry, which is, there's a lot of differences that could be among instruments, among laboratories, and so forth. So they've been spending, again, I don't know if it's been 10 years, but maybe it has, trying to get a handle on this and to really improve it. Here, the contact is Lily Wang. And so we're really looking at how do we have greater confidence and comparability among different instruments and within instruments across time. And there's all kinds of different fields where this is in the biosciences where it's important. So one of the goals, we want to have a measurement service. So if you have, beads that you think are good for flow cytometry, you can send them to us. We can give them reference values and send them back to you, which you could then be distributing and sharing. We are making reference materials ourselves for this purpose, designing assay protocols and associated infrastructure. And so we want to have materials and protocols that can support people with having good comparability among the results. So some of the ways that we've tried to do this is that there's a whole community that's come together. I'll show in the next slide some of the different stakeholders. There's a lot. From anywhere from manufacturers of the instruments to pharma companies and everywhere in between, other agencies across the US and internationally and so forth. So that's also a forum for discussing emergent issues. They've done 2 interlaboratory comparisons, and these are like monstrous, like 40 labs involved and 10 materials. And the amount of data is just crazy to analyze it, this whole workflow of how to handle that. And they're making progress. They have 5 SOPs that have been used in the inner labs, and things are improving year to year as this plugs along. And so lastly, here's a picture of some of the different organizations that have been involved. So you can see it's quite a lot. There's a complex structure, different teams. I'm not even going to get into that. But I think as this advances, hopefully some of these solutions can filter into the NAMS world for those that use flow cytometry to help increase the quality of those results. So with that, I'm happy to have any questions. Good.
Thanks. This type of measurement science work is so important to ensure reproducibility and reliability in the methods that we're all developing. Okay, so now I'm going to invite our other NIST colleagues to come over. I think Darwin will be first, right? Okay, to discuss organ on a chip standardization and research ongoing at NIST. Sure. To advance, and then if you want to use the pointer, you just got to go to the right.
Okay. All right. Thank you very much. Thanks for the invitation. Thanks, Elijah, for helping to that. Thanks, Nicole, and the rest of the team, and the rest of the team. Let me just put it here. Is that better? Okay, good. All right. So I just click on this. So yeah, I'm a project leader at NIST. I've been working doing research, but I also put myself in the middle doing things on standards since about 2016, specifically in microfluidic first. And then since 2021, I'm going to show a couple of things here. Then I've been trying to help the community on MPS. So I just want to share some of the things I'm involved and some of the people that I have interacted with. So first of all, the top line is the OTs of the Organ Tissue on a Chip Engineering and Efficacy Standards Working Group, and that's a group of stakeholders that I gathered a few years ago, and we've been trying to work towards the development of standards for MPS, specifically the engineering aspects of it. So we've also been interacting with the 3 years with the SCB, which works with the section that with MML at NIST, and with CAAT, with the Center for Alternative to Animal Testing at Johns Hopkins. And the main idea behind this working group is a group of people, right? So it's not a working group within ISO or anything like that, but we like to call it a working group, is to build the basis for publishing. So we wanted to start with some guidelines, which basically help us then to keep on the development towards the standards. And the aspects that we, as I mentioned, that we wanted to work on is on engineering of organ on a chip. And then we're trying to map out, we're still working on this. One of our first goals was the first couple of goals were to have a workshop, which we had last year, and a publication on what we wanted to do approximately. So along those lines, this is a bit of a history. As I mentioned, we started the group in 2021 in the summer. By the end of the summer, we had our first meeting. These were the first stakeholders and agencies that were into it. So you can see that we're working with the FDA as well, with Hyung Song, ATCC, Emulate, Hesperos, and also the university, the Michigan State University, as well as Sin Vivo. So when we started the discussion, try to figure out what we're going to do in terms of, so we're in a huge group, so we have to choose which organs we were going to start working with. So we chose heart, kidney, and liver. So later on, we finally got our website that is hosted by NIST. And so as I mentioned, we had the first workshop on this work last year at Michigan State. And then we had our first paper published in Lab on a Chip at the beginning of 2024. So this is how the website looked like. So we have some information in terms of the background and also the announcement about the workshop. We would like to use this website to start to put some of the work we have been doing, like on reports and things like that. This is basically trying to preach to the choir here. This is just a slide to show, how we are interacting with other stakeholders, and basically we're interacting with the FDA and with NIH, right? And of course, there are other agencies that we would like to interact with, and I think this is a great place to get to meet some of those people who are really into this. So I'm looking forward to talk more with you and others. So this is again, this is the ecosystem as I'm seeing it right now in terms of who we are interacting with. So, and this is coming because I am also the chair of the International MPS Society. Is the chair of the interest group on standardization. So as it turns out, we're going to have a workshop next month at the MPS World Summit. That's going to be on June 9th and 10th. It's sold out, which is good and bad for them to just know and might want to go. But so we're going to have over 100 people plus the speakers, and they are mainly from they're from all over the place, right? So there's going to be from the FDA, from CDRH and CBER. We're also going to have them from the European Union. There's a speaker from the European Commission, and then one from Netherlands. And we're going to have also speakers from Japan and Korea. So we're looking forward to have this happen and really get to hear what they really want to work on, and so that's very important to us. So anyway, this is just to show with whom we're really working with. And again, I want to expand this if it's really possible. So going back to what we're trying to do. So this is an example of some of the work we are doing. We're having our website for use of the people internally. The aspects we first thought on trying to look into for these organs. So this is how they actually look. I don't want to say much about it. So I just want to wrap up these parts. I'm just going to talk about this, and then I'm going to talk a little bit about the research that we're doing in my lab. So in terms of work that is in progress, as I mentioned, so we basically selected this working group, a steering committee, and we're working on a roadmap. And we're looking also into start with after the workshop of last year, one of the sub working groups, the one working with kidney, was working on getting all the terms that maybe will be useful for kidney on a chip. So we're going to try to finish this within this year. In terms of workshops, as I mentioned, so we're going to have the workshop at the MPS in Seattle. We're gonna have speakers from USA, Europe, and Asia. And then we're planning on setting up some sort of annual meeting, which I think would be great if we can talk with other agencies, so we make it really big and more inclusive. And then this is just to show once again that we're working with other agencies, mainly so far with the NIH. We just had an MOU with them to work in anything that has to do with workshops and NAMs and all that. As I mentioned too, we're working with the FDA, and we're looking forward with collaborating with other agencies. So going briefly and quickly through some of the work that my team is doing. So these are two images of, I mean, these are artistic ones, right? But it show overall what is the main idea of what we're doing. So we have a system that it's similar to a transwell system. For the ones who knows what that is, I guess everybody maybe here knows. That's great. Don't have to go through much of it. So we're having a channel on top and channel at the bottom with a membrane with pores right in the middle as a transwell. But the main difference here is that we've been able to microfabricate gold electrodes on both sides of the membrane. So we can use those electrodes as a manipulation tool. That means use it with for dielectrophoresis, so we can trap cells on both sides of the membranes and keep them there in a short time. And we can also use those electrodes for measurements of things like impedance or cell beating. So what that allows to do is to do real-time continuous measurements of the behavior of the cells within our systems. So, and this is a bit of that. What I'm trying to show here is what I just mentioned. I think it's easier if I can't really see it here. Anyways, so this is when you do the trapping, and I'm just going to show you a couple of videos of the work. So because we have electrodes on pairs of electrodes on both sides, we can actually do this. So this is an example. So what you're going to be seeing when I click is the trapping of cells. This is with a microscope that is an inverted microscope. So we're seeing the green cells here are the ones at the bottom. These are endothelial cells. The one at the top that you will see through the gaps between the electrodes are Hep2 cells. And so with this, I just want to show how we can quickly trap cells, and we're doing both at the same time on opposite sides of the membrane. So that should allow us to then start doing some of the experiments a bit faster. And then the other thing we can do with this after trapping, in this case, is not to measure, but to look at cells. And because we were able to trap both on opposite sides, we can also look at things like, for example, here, the endothelial cells are the red ones. So they are not forming the junctions, right? So they're really moving freely, but we can see the interaction between the cells from the opposite side through the pores, which are only 1 micron in diameter. And so we see how, for example, at some point you will see here that the cells are, so they're spread out, and how there are some interactions we believe are physical interactions through those pores, because even though they cannot go through, they can poke through. And let me go, how could I go back? Okay, let's see if I can run it. Sorry. There you go. So yeah, so what we can see, so they're spread out, and at some point they're going to start to open here. And I mean, we think we still haven't finished this work, but we want to figure out if there are some sort of interactions that are physical, in addition of maybe chemical, where they basically open. We think this might have some something to do also with cell extravasation. There's some things that could lead to that. So we'll keep on working on it. So, and then the last part is the hard on a chip part, where, so we have the same type of system. As I was saying, there are electrodes on opposite sides of the membrane. And so we have the system connected to PC boards to have a lot cleaner systems. So what we have here is the same type of system, but we customize it for these cells and for the type of experiments. And then what you can see here are the actual electrodes, and so this is a top view of set of electrodes on both sides of the membrane. So they're all aligned. We have cells that we basically I made in the lab these iPSCs, so we can differentiate them now into hard cells. So these are the markers for iPSCs, and then the markers for cardiac cells. And then this is an example of how they basically are beating. This is in the well plate, and this is fresh out of the oven over the weekend. The postdoc could show cells basically beating now on the device, so we can start using them. They're a bit slower on the actual device, but so they are very finicky about temperature. So, and then we're also working on that I don't have here, but fit for purpose systems in terms of the shape of the devices to look into shear stress and how. So I had this slide, but because this is going to be public, I took it out. So I was a bit concerned because we're still working on that. And with that, I just want to finish the presentation. Looks like I cannot move forward. Okay, so I just want to thank the people who are working in the lab, and if you have any questions.
Any questions here? Any questions in the room? I have one quick question for you. You're using iPSC models. Have you looked at variability among donors? Or are you not at that stage?
No, I'm not at that stage. This is a
Okay.
set of cells that I purchased, and then I induced them. I made them iPSC, so these are cells from the skin, right?
Okay.
So this is only one set of
Uh-huh.
cells about, yeah, this happened during COVID almost.
Yeah,
So
yeah.
I managed to get them all, but yeah, so no, we're still not working personally with other cells. I think once we get the system up and running with all the results that we can get out of these cells, then we're gonna jump into that.
Okay, great. Thank you.
Yeah, sure.
Okay, fantastic. And so now I will turn it over to our third colleague from NIST, Mandy Esch, who will talk to us about some additional NPS work.
Yeah, thank you so much. My name is Mandy Esch. I'm a project leader at NIST. And thank you to Elijah and to Darwin for sharing your time in the spotlight with me. So I'm part of the standards working group that Darwin is organizing. And so in order to develop standards, you really need to have good working knowledge of microphysiological systems. If you don't know how to use them, how to develop them, it's hard to develop standards that are really relevant to people and that people can use. So we have a small research program to develop and work with microphysiological systems. And so how do I advance? Just here. This one? Yeah. Okay. All right. So we have two kinds of works going on. We have single organ systems and we have multi-organ systems, and I'll talk about multi-organ systems in just a minute. But first of all, we have single organ systems, and we've developed a number of devices that can recirculate small amounts of liquid in a unidirectional way using gravity to drive the liquid flow in the device. So the special thing that these devices can do is they can work and recirculate small amounts of liquid, which is sometimes difficult to do with devices that have active pumps and active components. We've tested these devices with endothelial cells, but they can be applied to any types of barrier tissues. And we don't do all this work ourselves. We have, luckily, we have collaborations. And so the tissues that are in the devices can be, probed in the presence of shear. We can do transport experiments in the presence of flow, of course. and then also interactions of any cells or components, drugs with the endothelium or any other barrier tissue in the presence of flow. That it matters, show these data that I've put together for you. We found, for example, that bidirectional flow that sometimes is used with gravity driven devices sort of produce tissues that are more similar to a disease condition than a healthy condition. And so, so it matters that we have developed these devices and that we use unidirectional flow. Here you can see a comparison between unidirectional flow and bidirectional flow in terms of the IL-8 and IL-6 expression, meaning that there's more, you can see that there's more higher inflammatory conditions in the bidirectional flow conditions in these devices. So, so it matters, which is good to know. And we have established a couple of collaborations, one with Worcester University with Dr. Stewart, and one who's working on an infection on a chip. And we help with the growth of the endothelial cells under flow and within these microfluidic devices. And then we have a collaboration with Binghamton University, and there Dr. Mahler develops a kidney on a chip, and here the recirculation of small amounts of liquids is also of interest. So for multi-organ microphysiological systems, there can be many justifications or motivations to use multi-organ devices. For us, we have a very small program, so we have to focus. So we work on one device to look at estimating the bioavailability of drugs that are orally taken. And so this device has a gut component, a gut chip, and a liver chip that are brought together in this housing. The tissues can be cultured separately, so it's a modular device. And then you can put them, once the tissues are mature, you can put the two chips together. And then there are two fluidic loops, one representing the systemic circulation within the body, and the other one is the digestion loop, where basically you take your drug. And so the question is, how good can a device like that simulate or recapitulate the oral uptake and then later the bioavailability of a drug? And so we test this system with different drug concentrations. We build PBPK models for the system to see what kinds of data we can expect. And then we try to do an in vitro to in vivo conversion to see how well the data compare to in-human data and what one must do in order to extrapolate the data that we obtain with the devices. And so because the devices will never, there are always compromises that we make. They will never be the same as human body, right? So there must be a way to do the in vitro to in vivo conversion. And so we want to learn about how do we go about that? And the second motivation is to measure the primary and secondary drug toxicity of drugs. And I think here in particular, multi-organ microphysiological systems have their potential, in particular in looking at secondary drug toxicity. Because primary drug toxicity you can test on single tissues, but with multi-organ devices, you can, or a tissue can metabolize a drug, create a toxic metabolite, and then it can recirculate to other parts of the system. And so I think the tissues have the potential to detect secondary drug toxicity, which is hard to detect, and sometimes at fault for a drug not doing well in a clinical trial. And so a question that I'm asking myself, when we do this research, how well will microphysiological systems do, maybe by themselves and maybe in combination with animal experiments? So those are some motivating questions. Now, for developing standards, it's good to know all the parameters that can affect the experimental data that we get with microphysiological systems, and there are many. The devices themselves, how are they created? How are they designed? What design philosophy do they follow? What are the scaling factors, tissue volumes, and so on? Then the tissues, of course, can be created from different types of cells, have different densities. The medium composition plays a role, and then the operational parameters as well. How often do we change the medium? How much of the medium do we change? How many days do we operate the devices? So there are many, many parameters that affect the outcome of these experiments. And for me, particularly important is the liquid volume in the device, because I want these devices to do well when trying to detect secondary drug toxicity. So if a device has too much liquid in it, then any toxic metabolites will be diluted and you won't be able to detect the toxicity as well as if the device has, near physiological amounts of liquid. And so the right graphs are a simulation, a mathematical calculation of that. And you can see that the metabolite concentrations indeed are much lower when you have more liquid in the device. Makes sense. So we've developed, this device is called the Body Cube. It's also a modular device, and you can bring together different organs. It uses gravity to drive liquid flow, and it's a multi-organ device. And basically, this device allows us to operate with near physiological amounts of liquid. So we can load this device with all kinds of volumes of liquid, but also with very low amounts of liquid. So in this device is scaled 1 to 73,000, and the liquid volume that we operated with is 80 microliters, or larger. It can be larger too, but the minimum is 80 microliters. So yeah, this is just how it's operated. It rocks back and forth on a rocking platform. And so that allows us to investigate all these different parameters, but in particular, what role does the amount of liquid play in the devices when generating the data? And we've moved forward from the Body Cube and done a few interviews with industry, and I found out, no surprise, that the industry is particularly interested in liver heart, liver kidney, and liver brain combinations. And so we've developed a new device shown here, one for a rotating platform and one for a rocking platform. And we'll be doing experiments with those. So they're forthcoming. Yeah, so just to summarize, we work, basically we do technology development, which helps with the tech transfer. So companies can license our patents, and that's part of the NIST mission to support the US industry. And then we do experimentation with the devices, and that helps with the standards development. And then we also work on the in vitro to in vivo conversion, and to see, what kind of impact can the devices have? Can they be really so good that, we can use the data reliably at some point? And with that, thank you for your attention.
Do we have any questions in the room?
Thanks a lot. That was really interesting. But do you measure oxygen saturation in the media at all? I mean, that's pretty important for physiology.
Sorry. Yeah, at the moment we don't, but I think it's a good idea and we should. Yes. Yeah. Thank you.
I have a quick question to try and equilibrate what you said at the beginning about the effect of unidirectional flow versus bidirectional flow, and then the device you described at the end, which is the flow is being done by gravity and it's rocking. So does that mean that it's more representative of a disease state? because you don't have that unidirectional flow in that body cube?
Yeah, so I have to say, a presentation is always kind of linear. So originally we did the experiments to look at bidirectional flow and the development of the cube at the same time. And at the time I was very excited about the cube. And so we developed it, and yes, it has bidirectional flow. The disease state is really relevant for the endothelium.
Sure.
If you have the endothelium in the cube, yes, it will be more representative of a disease state. If you don't have it in the cube, regular tissues like liver tissue, non-barrier tissues, don't, well, respond badly to any kind of shear. So if the shear is very low, even bidirectional, it should be okay. If it's high, bidirectional or unidirectional, it's bad for the tissue. Now, having said that, because of this bidirectional flow in the cube, I think it's a drawback of that particular technology. I've developed the next generation, which I showed, like, I think in the last slide or the third to last slide, which is a new device that we're working with. And that one does create unidirectional flow and has
More of a microfluidic.
Yeah, it's well, both are microfluidic devices, but this one, maybe I can go back. This one sometimes it's it's this one right here. Yeah, it's this one right here. So it's kind of unique. It has unique features, but at the end of the day, it can be operated with very low amounts of liquid as well, and it does have unidirectional flow. It's an open device, and it accomplishes that because it doesn't have to be filled everywhere at the same time. So some, typically in a microfluidic system, if it's closed and you don't fill it at some point, there's an air bubble, right? And if an air bubble goes over your tissue, the tissue will die, right? That's the end of it. So in this device, that's different. It's not closed, so the air bubble won't move over the tissue. But not all parts of the system have to be filled with liquid at the same time. That's basically what it comes down to. I'm not sure if that.
It's really interesting. I have a lot more questions, but we're running low on time, so we'll talk more.
Thank you so much.
Thank you so much. That was fantastic. Okay. All right, so last and certainly not least, we have our colleague from the US EPA, Charles Kovach, who is the US National Coordinator to the OECD Test Guidelines Program, and he will be speaking to us about some updates from the OECD side.
Great, thanks, Nicole. Thanks, everybody, for hanging on until this part of the day. Charles Kovach, I've met some of you, but for those who've not, Charles Kovach, I'm the US National Coordinator to the OECD Test Guideline Program. So it's representing the US at the OECD, even though my home office is the US EPA's Office of Chemical Safety, Pollution Prevention. And I think it's kind of interesting actually to be the last speaker for today because, it's always exciting to see all the research and perhaps research that we're going to see moving through the test guideline process. And so that's quite exciting. It's always nice to know or remind in some cases what the OECD is. The Organization for Economic Cooperation and Development, founded in 1961. Kind of interesting that it's economically driven. It was developed or created rather following World War II. You had the Marshall Plan that was helping with the revitalization of Europe. Then you have the OECD formed where Europe was recognizing the need to continue to collaborate for their economic vitality. The 38 member countries currently covering a wide range of areas broader than just chemical safety. So tax and waste and energy and trade, education, finance, agriculture, fishing, a lot of areas. The area where we work is the Chemicals and Biotechnology Committee, which not surprisingly oversees work on chemical safety and biosafety through 11 different working parties or committees. Working parties is the term they use. And the CBC is what's, I guess you could say the most important thing that they have is this, for us, is the mutual acceptance of data. It's built on two principles of the test guidelines and good laboratory practice, right? So in essence, you have, it's the member countries and anybody who's an adherent to MAD, right? So you can do a single test and submit that data to regulators, and it would be received, right? Rather than undertaking that and undertaking it or conducting it multiple times, right? The working party for the national coordinators of the test guideline program, WNT, that's the mechanism by which, these methods are reviewed and through unanimous consent approved to become a test guideline. So that's where I'm the national coordinator, and Nicole operates as a co-national coordinator representing the US at this meeting to ensure our priorities are conveyed. Next slide. So today I'll just be providing an update of what happened at the WNT meeting. Again, you saw the OECD has a lot of activities. I'm just covering the WNT. So we recently held a meeting, a hybrid, they've all been hybrid, right? Back in April. I was in person. Nicole was participating virtually. What I'll be talking about today are some of the highlights. We approved 9 test guidelines or guidance documents. We added 18 new projects to the work plan, and then and I'll get into where the US was a participant on those. And then just to clarify, so some of these projects go through a series of development steps, and when they reach that completion, that's when they are approved, right? And then the new work is that work that's going to begin, and so that's where approving versus adding the projects. So these are the items that were approved, and so of those, there are 3 new test guidelines, 3 revisions or updates to existing text guidelines, and then 3 that are, one's a new guidance document and 2 of them are updates to existing guidance documents. The asterisk, which I'm going to show you in the next slide, indicates that it's NAMS related. I think it's kind of neat. We start highlighting and focusing where those are occurring. And then there's a plus here, that was the US project. So these are the NAMS approved test guidelines, right? The first one, the rapid estrogen activity in vitro assay, the reactive. So this screening assay, it adds a new endpoint, reproductive endpoint through this, through the I don't know how they say it, the GFP genetic construct, right? Adding that new endpoint for expression, right? The second one that was added is updating to test guideline 442D, adding a new method, the EpiSense-A, for inclusion or in vitro skin sensitization. The next one, updating the test guideline 467, including defined approach for eye hazard identification. So previously, about a year or 2 ago, we had approved the defined approach on eye hazard identification for liquids, right? So now we're adding that component for solids. You can see including the bovine corneal opacity, permeability, laser light based opacitometer, right? And then last but not least is the updating test guideline 496, including the OptiSafe macro molecular test method for eye hazard potential, right? So, here, we call this a Me Too in the OECD test guideline space. So they're adding this onto that existing test guideline, and Of course, we're very proud of this when it was introduced a few years ago, and here it is now being approved, providing accurate, high sensitivity, non-animal eye irritation test, right? Newly added projects. There are 18 that were added to the work plan this year. The first three, I'm just trying to show you how they're grouped in a way. These are on, biotic systems. Then the next one, which is in the red category, there is they categorize them. It's degradation. And then the other 14 are health effects, right? So just showing you, this is kind of a theme, what we're seeing with distribution of new and incoming projects that are coming through the OECD. There are more and more appearing on health effects. It's interesting, I think, to note the first three are all on honeybees, right? Pollinators, right? So you can see where the EU is going with a lot of their attention right now. Not that we're not doing that in the US, but I just think it's kind of interesting that we were driving three new projects related to honeybee or pollinator sensitivities. So I'm going to talk about the US projects that are in yellow highlight here. Right, so I think the first US project is an update, and again, another Me Too method onto existing test guideline 442C. So looking at the electrophilic allergen screening assay for inclusion. The next one is a validation of the in vitro micronucleus assay for engineered nanomaterials. A lot of activity in the EU on nanomaterials. They have kind of like a consolidated driving initiative, if you will. The US is a participant into that project. There are many projects but on this one in particular, US is a participant. A detailed review paper on the application of error-corrected next-generation DNA sequencing for gene mutation evaluation. US leading this project. Actually, I think this is Emily, and no, that's not you. What's that? Oh, the ESA is yours. No, this one. It's still, yeah. Thank you. So anyway, it's developing a detailed review paper and conducting a retrospective performance assessment on, like how well it has been performing on this assay. A revision to an in vivo genotoxicity test guideline. So what's kind of interesting about this one is they're evaluating, or in the sections of evaluating and interpretation of results sections, it's looking at, how to. There isn't much guidance on how to, or there's poor guidance on how to assess whether response is positive or negative. And they want to introduce a new concept that utilizes historical negative control distributions on data. And what's interesting here is that's going to potentially apply broader than just genotoxicity data, right? And I think it even introduces opportunities for in vitro data later. And then lastly, being led by the US EPA, it's a new guideline for testing oral, dermal, inhalation, and injection toxicity pathways, microbial pesticides. So essentially taking some existing and well-used and documented EPA guidelines and placing them into the OECD test guideline process, right? Making them available to a wider audience. At the WNT meeting, so the prime part of the meeting is approving test guidelines and identifying new projects to add to the work plan. There are some other updates and activities. A few highlights for you. The first one is a stakeholder workshop on operational financial aspects of validation. You probably heard me talk about this before. And so some of the highlights of this project started in January, well, actually before January, but in January 2023, the OECD published what's called a call for mobilization of resources. So essentially, they're trying to elevate or raise the awareness on the importance of resources for validation, right? and with that call, kind of ignited, if you will, activity within or paralleled with, complemented rather, the WNT. We organized in the US. A few of us were very active in, helping to scope and hold a workshop, some webinars in November of 2023. You can see those on using this link, looking at overview of just kind of like an overview, broad overview of what validation was, looking at method readiness and a number of case examples. And then we hosted a webinar, we hosted a workshop in December, and that had its intention. We had a survey ahead of time trying to see where folks are, what were their interests. We had a diverse group of researchers and developers and trying to capture that whole spectrum from research and development to agency implementation and even financial organizations, trying to share that dialogue to identify what are some of the lessons learned and good practices that can go forward. Some of the recommendations that we had distilled down. So financially, it's kind of like guidance or best practices on detailed budget planning, how to announce those or share those priority areas for funding, how to encourage method readiness filter so that maybe you can identify projects using the term shovel ready, but these aren't really shovel ready, but you get the idea. And encouraging earmarking budgets for validation, specifically for NAMs, right? So many times budgets are identified for development, but maybe not specifically for validation. On the organizational side, some of the things that came forward, so guidance document 34 is that foundational document for the validation of methods, right? And so what we recognize is there is a need for an online companion to that talks about preparing SOPs or identifying partner labs or a lot of these other areas that guidance document 34 doesn't necessarily satisfy. Also, something that came up a lot was how to connect method developers and available resources, and then filling the communication, those gaps of communication, training, and education. We will be, the WNT, the Secretariat will be publishing a report expected in June, and it'll be, again, all these materials are available at that link. And I want to give kudos out to all the ICFAM folks who participated to help publish the validation, qualification, regulatory acceptance, new approach methodologies, because that document and the activities that we do here really informed the US position in these discussions. And it makes us a leader in discussions because we have real examples of where we've already done many of these things, through the ICFAM roadmap. We are already working on, connecting developers to end users, try to, encourage some of those prioritizations of methods, aligning budgets, and some of these other things. So it's kind of like that was a really helpful to have handy during those discussions, and a kudos to you guys. I'd mentioned this, so the project to revise guidance document 34. Again, you guys have heard me talk about it before. The WNT initiated activities to modernize that process back in 2022, and there are a number of webinars that you can click on a link to revisit those. In 2023, WNT formally approved that project, placed on a work plan where the US, as well with the European Union and the Netherlands, are co-leading that project. And, hats off to Allison, Warren, and Nicole, who are really instrumental in participating and guiding and leading those discussions. Over the past year, we've developed the expert group, participated in various meetings, and we've held meetings with our national coordinators to discuss guidance document 34 and make sure folks are clear on, what needs to be addressed and what's that direction that we're going to take to do it. And so right now, we've been talking about and identifying areas within what's section 4 of the document on the design and conduct of validation studies. So looking at identifying laboratories and looking at different steps within the validation process and looking at reference chemicals and reporting of data and recording of data. So there's been a lot of areas that they're touching on in section 4. And then moving into, we're starting to explore topics like readiness criteria, what are some of the different things to include in defined approaches which aren't really articulated within the existing version of guidance document 34. That's, it's a 20-year-old document, and transferability. And so we will be meeting again as a WNT sometime later this year. And then what's kind of neat, and I told you there were other things that were discussed at the WNT meeting. And what's nice is that, we learned about the Nano Harmony project. It's a European funded project to support the development of test guidelines and guidance documents, specifically for nanomaterials. However, what's really timely about this project is it offers, a framework and multiple entry points for the public, for the government agencies, for developers, for funding organizations to engage or understand maybe where something is or where they can contribute. So I think it's very timely, especially as we're talking about guidance document 34, revisions of guidance document 34, and looking at, some of the outcomes of and recommended actions for the stakeholder workshop. And then here's a really nice project or product. So and invite you to take a look at that. My last 2 points are upcoming dates. If you have an idea and you want to submit it as a proposal, everything shifted up a few weeks this year. Oftentimes it's November. This year will be October. So that might light a little fire if you're thinking about it. And an FYI, because the date for the WNT meeting is a little earlier next year in 2025. So with that, I'll take any questions.
All right, thank you, Charles. First question, when will accepted test guidelines and other documents be available to the public, and what's the best way to find out about these?
Yep, good question. So typically, so remember I mentioned that the WNT is one of the 11 parties under the CBC, the Chemical Biosafety Committee. They have their meeting in June when they will finalize all the materials and then make them available. So those will likely be posted to the website in July. If you're on the ICVAM distribution list, that's something that I can, that I will make an announcement at least through that email. Otherwise, you just have to kind of check back on your own.
Well, and we'll put it out through NICEDM News as well. So I'm assuming pretty much everybody who's online right now or in the room is subscribed to NICEDM News.
Excellent. Yes, thank you.
All right, we have 2 more. For the revision of the in vivo gene tox test guidelines, can you comment on which test guidelines are being considered? Are all in vivo gene tox test guidelines included? Is there any applicability of this type of analysis to the in vitro test guidelines?
So I think you're referring to the project that was recently added to the work plan. I don't have that in front of me, so I can't tell you specifically which ones, but I think that they're probably going to explore that. I know they have a few in mind. If you contact me, I can at least tell you what the proposal said and then put you in touch with the actual researchers who are leading the project.
Thank you. And then Nicole, I'm going to actually toss this one to you because I believe it's for the ICFAM validation report. Does it provide guidance on how to evaluate the proficiency of such as a contract lab submitting data to the agency regarding a given assay? In other words, an assay may be validated, but how do we know the lab in question does a good job of performing or deploying it?
Yeah, that's a complicated question. I will say that one of the, I think, really valuable things about the validation work group report is the inclusion of a lot of information from our NIST colleagues on things like application of measurement science and control measurements and doing variability analyses. And there was a publication, a Peterson et al. Publication that formed the basis for a lot of the information that went into the VWG report. And the VWG report has like a pretty extensive appendix that includes lots of guidance and recommendations around things like data reporting, good in vitro laboratory practices, statistical analysis methods, et cetera. So there, from a sort of quality control and quality assurance standpoint, there is a lot of guidance in there for labs specifically. In terms of acceptability for a particular regulatory authority, the guidance that you're going to find in the validation report is the same as the guidance that you're going to get from any individual authority, which is come talk to us. So establish lines of communication early in the process with the regulatory body or the agency that you're interested in doing an evaluation of your data or your capabilities. And tomorrow, Emily and I will both talk about follow-up efforts that we have in that space to establish better lines of communication between method developers and laboratories and stakeholders, both in the regulatory decision-making and the regulated industry fields. Okay, thanks, Charles. Okay, and now I will invite Dr. Helena Hogberg up here, who's the Deputy Director of NICEDM, to do a wrap up of this wonderful afternoon that we have had and give us some feedback and advice on preparing for tomorrow.
Yeah, thank you. So I'm going to be really, really brief because I know we're already over the time, but what a super interesting 2024 first day of public forum. So I'm looking very much forward to tomorrow, which is going to be even a longer day. So we're going to be here in the same room tomorrow, but at 9 o'clock we're starting. Make sure you get some extra time to get through the security tomorrow because it's a busy time at 9 o'clock. There's a lot of people coming in. At least 30 minutes. And if you're going to spin around like I did today, you probably want to have 1.5 hours. But I hope you know where you are now today. So we have a lot of public statements. That's going to start with tomorrow. We have 12, which is a record. It's amazing. But I just wanted to reflect a little bit back to today, just a couple of things. I thought it was really nice to see so many different ways. So we saw very physiological relevant and more complex microphysiological systems and new NAMs that have been developed, but also looking at new ways of looking at confidence in using NAMs. So for example, looking at human samples and try to bridge back. And then also the flexibility in using them for regulatory applications. So with that, I want to thank all the speakers. I want to thank the co-chairs of ICFAM, so Natalia and Susie. I want to thank the director of NICEDM and the executive director of ICFAM, Nicole. I want to thank all our innovative people that have been, they are the contractors to NICEDM, and they have been working behind the scenes and really put all of these things together. So thank you to all of them. And of course to the AV that are sitting in NIHS and in Raleigh. Thank you, Nathan and Chris and the team. And all of you, thank you for being here. Thank you to those online that still are on there. And I'm really looking forward to see you tomorrow and hear more about NAMs.